And hopefully as everybody else gets the notification and the information on how to get in, we can then get started on the lecture. So I am almost done grading your exams. It was truly an interesting mix of scores. I would appreciate feedback from you guys on what you thought of the process and what could be done to improve it. Because again, this is all new, so I want to try to make this, again, the goal of an instructor is to make sure that the test does not get in the way of you guys showing me that you have the information. And I'm concerned that this format isn't very successful at doing that. So I would appreciate some feedback from you guys on what you think the best way of being able to do that kind of stuff would be. So I'll do that. I hope to have the grades done tonight. And then in all likelihood, based on what I've seen so far, we'll be curving it to some extent. And I'm not 100% sure how to do that on Canvas. What I may just end up having to do is add a second grading assignment where I add the additional points you would get from a curve into it, because the way that those proctorial tests are set up, I can't change the point value. Other than, I guess, going in and putting in fudge points, but it's the same way, and it'd be faster just to do a second item. So we'll see how that goes. I'll take a look at that, and we'll have that done. So I'm hoping to have those posted to you guys by tomorrow at some point. And then we have a quick turnaround. We have 3 lectures and then another exam. So the good news is it is just one organ system, so it's just the digestive system. So it is a smaller bit of information. But it's also a little bit of time, and it's going to go quick. We have that, and then we have the reproductive system, and we're done. So 2 quick systems with a lot of content in them, and then we'll be done with that. Only 2 assignments for this section, a unit review and a Physio-X. The Physio-X is going to be especially important because that's the wet lab that we would be doing in the classroom, where we would be, digesting our proteins and digesting our carbohydrates and looking at those results and understanding those mechanisms that are taking place inside of our stomach. So that's going to be really, really important. And then, like I said, the format for the exams, I'm assuming, will be similar, if not identical, to what we had for this last one. So that's, like I said, I appreciate your feedback. And once we look at the final numbers, we'll see what we can do for that. All right. Any questions on any of that? The other thing that I'll point out to you is there is a lot of histology on this exam. This is an exam that is typically a PowerPoint presentation because there's a lot of histology on it. The histology on that CRC visual anatomy is pretty good. They've got a lot of stuff. There's some other things that we will add to it as well, and I'll try to add more resources. But make sure you're also taking advantage of your textbook, your histology atlas, the anatomy lab that we have on has some decent histology. So make sure you're taking advantage of those resources as well. All right, we have a pretty good number of students now. Again, we've had a mass exodus, as would be expected in this kind of a situation. In the month of April, we've lost a lot of students, but again, this is kind of a crazy time and a crazy learning environment. So I appreciate all of you that are still here. I hope all of you are safe and taking care of yourselves and your families and everything as well. And let's do our last a little bit of push towards the end of this class. All right, so again, here is our format. We're going to be talking for the next 3 lectures on the digestive system, talking some anatomy. Again, even when we're in the classroom, I don't have the time to go over all the anatomy. There is more of an onus on you to be able to take advantage of that and learn that on your own, and the assignments and the lab exams. So let's move and switch to the lecture and go from there. So, as I mentioned, our goal is to talk about the digestive system. We are going to follow the mystical and magical journey that your cheeseburger that you had for breakfast this morning has taken on its way. Like all organ systems, we have 2 main, or like many of our organ systems, we have 2 main groups of organs. We have the primary organs that are responsible for the major functions of that organ system. And then we have the accessory structures, the accessory organs that help and assist in that. In the case of the digestive system, our main muscle group, our main organs of the digestive system collectively form what we call the gastrointestinal tract. It is a hollow tube that runs from mouth to anus, and in a cadaver is about 30 feet long. Of course, as you remember from 430, hopefully when you got a chance to look at the cadaver, the reason we specify in the cadaver is because while death is many things, one of the things it is very relaxing. And so the digestive system has a tremendous amount of muscle tone in it. When we pass away, that muscle tone goes away, the muscles relax, and the digestive system extends. So all of you have digestive systems, have gastrointestinal tracts or alimentary canals. Those two terms are interchangeable. But yours, because you're alive, is much, much shorter. The other set of organs, the ones that assist in that process, are what we call the accessory organs. One of the easy ways to tell the difference between the primary organs and the accessory organs are, as I said, the primary organs form that alimentary canal, form that muscular tube that goes from mouth to anus. So again, if food goes through the organ, then it is considered a main organ. All right, so the mouth, the esophagus, right, the stomach, the small intestine, the large intestine, right, things like that are things that food passes through. Right, the liver, the spleen, the gallbladder, the pancreas, those are examples of accessory organs. They provide important and in some cases vital functions, but food doesn't pass between them. Right. Your teeth and your tongue are accessory organs, right? Food does not pass through them. I mean, I guess if you have a large gauge piercing in your tongue, technically food can pass through it, right? Food may pass between your teeth or over your teeth or under your teeth or over your tongue, but they don't go through them. Right, so those are, again, I guess if you have a really large cavity, food could pass through your teeth. But for the most part, right, those are things that are accessory organs. Food does not pass through them. They go by them, but they do not go through them. As we often do when we are talking about an organ systems, one of the first things we do is we talk about our functions. And for digestive, there are 6 basic functions. These functions are numbered, as you can see here, but this is not necessarily sequential, right? This is not a sequential list. That's tiny. Let me make that a little bigger. There we go. That is not a sequential list. Now, obviously, some of these things have to happen first, right? Ingestion obviously has to happen before all of the rest of these processes can occur. Typically, defecation is going to be the last part of this process. So again, there is some sequential nature to it, but it isn't like we do all of the propulsion, then we do all of the mechanical digestion, then we do all of the chemical digestion, and so on and so forth in that fashion. Many of these processes occur in multiple locations. In fact, for instance, propulsion obviously has to occur in all of the organs. Otherwise, the food stays where it is, right? If it's going to move from one organ or through an organ, it's got to be propulsed. Absorption can occur in many places, including your mouth. So there are many locations where absorption can occur. And the other important thing to remember about absorption is how we get the substance into our body. Right? The key with absorption is where we pass through membranes to enter the body. One of the interesting things about the digestive system is that much of the function that occurs in it occurs outside the body. Right? We've talked about this in the past. I think if I change the slide, I think I lose this writing that I've put here. I guess none of it here is really that important. Here, let's do it there. I'll sneak it up here in the top. If you remember, and we've talked about this before, as I just mentioned, our alimentary canal, I wonder if I can make that longer, larger, excellent, is a big hollow tube, as we said, that goes from mouth to anus. And as we have also talked about, the lumen of that is outside the body. Right? If I take a penny and I put it in my hand, lay it on the palm of my hand, it does not enter my body. If I wrap my fingers around it to form a fist around that penny, my body is now surrounding that penny. But again, that penny has not entered my body. To get that penny into my body, I have to pass a membrane. So I have to sharpen the edge of it and stab it through the skin of my hand into my hand, and at which point it would be inside my body. And it's the same thing if I put it on my tongue. If I lay that penny on my tongue, it has not entered my body, right? If I stick my tongue out and put a penny on it, it has not entered my body. If I bring my tongue back into my mouth and close my mouth, again, my body is surrounding the penny, but it is not actually entered into my body. That penny enters the mouth. If I were to swallow it, it would pass through the entire alimentary canal and come out the other side through the anus, and it would never have entered my body because the lumen is considered outside the body, and we, of course, have a fancy word for that. It's the topological exterior. All right, so we have that penny. That penny enters into my alimentary canal. It passes in one side, comes out the other, a little cleaner or a little dirtier, depending on how you think of it. But if it were to enter the body, what would have to happen is that it would have to pass the membrane of the wall. And once it passed the membrane of the wall, it would then be inside. Right? We have no currency receptors. We have no pumps. Our body has no need of money on the inside, so it passes right on through. Whereas that cheeseburger we break it down, and once we break that cheeseburger you have for breakfast down, then it is absorbed through the membranes and finally enters into the body. And that's the other key point that I want to make here. When we look at the breakdown of the food, notice there are two different types of digestion, right? Digestion is, of course, what we would consider the breakdown of the food, but there are two ways we break it down. We break it down mechanically, and we break it down chemically. A mechanical digestion is simply just the physical process of making something big into smaller things. If I have a boulder and I have a hammer and a chisel, I can sit there with that hammer and chisel and turn that one boulder into a bunch of tiny little pebbles. All right, I can break the one big thing into a bunch of smaller things. So from one boulder to 1,000 little rocks. But I haven't changed the composition of that. The rock is still the rock, right? Chemical digestion, on the other hand, is the process of breaking bonds. Right? This is where we are not just taking big fats and making them smaller fats. This is where we make that fat and we break it down into glycerols or alcohols and fatty acids. This is not where we turn big proteins into little proteins. This is where we break the bonds to turn those proteins into small clusters of amino acids or even single amino acids. Right? We turn polysaccharides into di and monosaccharides. This is where we break bonds. And of course, if we're breaking bonds, what are we going to always use for that?
ATP.
Enzymes.
True, absolutely. We definitely often will use ATP, but in particular, when we're breaking bonds, we are going to use enzymes. right? Enzymes are how we break it down. As we talked about in 430, you never like to use terms like never. You never like to use terms like always in an anatomy and physiology class. But as I said, every single chemical reaction that takes place in the human body, we have found an enzyme that helps to facilitate it. So anytime we're going to break bonds, and of course for breaking bonds, we're using hydrolytic enzymes to break those bonds and to break those larger structures into their smaller base components. Right? So again, it's not just a matter of taking that cheeseburger and using your teeth and grinding it up into smaller pieces. That would be mechanical digestion. But then we have to use enzymes to break down, like I said, those fats down into alcohols and fatty acids, those proteins down to amino acids, and so on and so forth. All right, so these are the 6 processes that are involved in digestion. And if there was a 7th, and again, it is going to be strongly related to all of the rest of this, that 7th would be secretion. Right? Because obviously much of the chemical digestion, because again, remember this is occurring outside of the body, we have to release fluids and enzymes into the lumen for that mechanical and that chemical digestion to take place. So if there was a 7th, we would call it secretion, although really secretion falls under chemical digestion and mechanical digestion. So both of those involve secretion. All righty, questions on that? All righty, excellent. Let's move forward. Got to clear the board. Excellent. So like I said, obviously some of these processes occur in just one location. Ingestion just occurs in the oral cavity. As we just finished talking about with the respiratory system, technically you can breathe air in through your mouth, but it doesn't make your mouth a part of the digestive system. I mean, the respiratory system. Technically, you could put a straw up your nose and you could get milk into your stomach through your straw. I know we're all bored at home right now, bored in our houses, but I'm not encouraging anybody to do that. So theoretically, while you could get milk through your nose into your stomach, again, it doesn't make it part of the digestive system. So ingestion occurs in just one location. Hopefully defecation just occurs in one location, right? But as I said, many of these processes occur throughout the digestive system. So in our mouth, many of these processes take place. We've got mechanical digestion that's occurring in our mouth. It occurs more in our stomach. It occurs in our small intestine. We're going to have chemical digestion occurring in multiple locations. So many of these processes are going to occur multiple times in different locations and in different ways as well. So that is going to be an important distinction we're going to want to make as well. All right. Now, the other important thing we need to understand is, again, this physiology and how it is controlled. As we talked about, the lumen of our digestive system is outside of the body. So it's a bit of a unique situation where we are regulating the environment that is outside of our body. Sorry, hold on, I'm playing around with my screen to make it more feasible for me. As we talked about, the lumen of our gastrointestinal tract or alimentary canal, remember those terms are interchangeable, is the topological exterior of the body. And so there are basically some major basic digestive processes that are going to occur. Inside of those organs, there is going to be some type of stimulus. And typically that stimulus is either mechanical or chemical stimuli. One example, when you have that cheeseburger for breakfast, is that the increase in mass in your stomach causes your stomach to stretch. That stretch is a mechanical stimulus. And as we know, we have stretch receptors that can perceive that stretch. And when they perceive that stretch, that is going to stimulate some response. But there are also chemical stimuli as well. Things like the change in pH, the presence of certain building blocks, like for instance, glucose or amino acids or sugars or things along those lines. So there are chemical and mechanical stimuli that are going to trigger events in our alimentary canal. So again, it could be the osmolarity, it could be the stretch, pH, substrates. And when that stimulus occurs, our organs of the alimentary canal are going to respond in two ways. They are going to either increase or decrease their glandular activity, and they are going to change the behavior of their smooth muscle, increasing or decreasing the contractions of that to increase or decrease the motility. All right. So again, our stomach stretches. As a response to that stimulus, two things happen. It starts producing more gastric juice, so we get an increase in glandular activity, and our stomach starts churning more. We get an increase in smooth muscle activity, and actually a change in the activity that's occurring there as well. Excuse me, too much grading this weekend. All righty. Let's talk about how these are controlled. Not surprisingly, when we're talking about glands, when we're talking about smooth muscle, those are all things that are controlled by our autonomic nervous system. Actually, let's do it this way. We're controlling smooth muscle and glands. And there are actually two neural mechanisms that we are going to use to control them. The first is actually the organs themselves. We have what is known as, where's my type? Here it is, what we call the enteric plexus. Plexus is a term we used back in the nervous system in 430. I don't remember if we've used it here, although I think we used it for when we were talking about the anastomoses in our cardiovascular system. We talked about how those are similar to a plexus, where you basically have this elaborate network of nerves. And these elaborate network of nerves are actually capable of making very simple decisions. So as I just finished talking about, sorry about that. Oh. My daughter's high school always likes to have the automated messages that come in. All right. So the stomach and many of the organs of the digestive system have these elaborate networks of nerves that are actually capable of making basic decisions. So that example I used before, where you have that cheeseburger goes down to your stomach and your stomach stretches. That stretch signal actually goes to this elaborate network of nerves that is actually in the stomach. And in the stomach, it says, okay, I've been stretched. I need to respond to that. I need to start churning that food. I need to start producing gastric juice. And that whole entire decision actually occurs right there in the stomach. So it is what we think of as we talked about this a little bit when we were talking about urinary system. This is our short reflex, right? Where it is an internal decision with a local effect. Right? And that is what this enteric nervous system is. This enteric nervous system, and what is affectionately referred to as our gut brain, right? Your gut brain is where our digestive system is able to basically make its own decisions. And now that I've gotten that over there, let's do this. So again, it is these short reflexes. These are internal, intrinsic decisions that are made outside of the central nervous system, right? This is all outside of the central nervous system. And it is that elaborate network of nerves that is used to make that decision. Right? For these short reflexes, again, like all processes, we have a stimulus. That stimulus then has a receptor. That A receptor then has a command center, and that command center then sends a signal to an effector, right? When we talked about our process of homeostasis, those are basically the four components that we have talked about. In this case, with these short reflexes, the stimulus has to occur inside the organ. So the stimulus occurs inside the organ. And the other big, I'm going to have to move this. I'll put it up here for right now. The other key component to this short reflex is that the effect is going to be local. So if it is the stomach that stretches, it is the stomach that is going to respond. So typically with this local effect, whatever organ was stimulated is the organ that responds. That is the key to these short reflexes. They have to occur inside of the organ, and they typically only, no, let me rephrase that, and they only affect the organ that was stimulated. Those are our short reflexes. Now, of course, the stretching of the stomach is going to affect other organs as well. But that ability to affect other organs outside the stomach require our long reflexes. Our long reflexes, which are our extrinsic controls, these have to pass through the central nervous system. Right? And of course, remember that means brain or spinal cord. Some of these are going to be spinal reflexes. This is going to use our sympathetic and parasympathetic pathways that we learned in 430, and we will talk about more again in this class as well. And notice also the key to an extrinsic control is that the stimulus can occur inside or outside the gastrointestinal tract. Let's go back to our stomach. You have that cheeseburger for breakfast, it stretches the stomach, and your stomach starts producing gastric juice. It starts churning as a result of it. But you could also be sitting there watching TV, and as you're sitting there watching TV, a Carl's Jr. commercial could come on. No hamburger in the history of mankind has ever looked better than a Carl's Jr. hamburger on the commercial. I'm not talking about in real life, but those hamburgers on the commercial look amazing. And as you sit there looking at it, right, is there the potential from that, just by looking at that, enough to potentially get your stomach to start producing acid and start getting a little rumbly? Or maybe your next door neighbor, right? Because we're all home and we're all doing our home cooking. They have that tri-tip that they put that nice dry rub on. They let it sit and marinate for 3 days in the back of their refrigerator. Then they brought it up to room temperature, put it on that hot grill to sear it, moved it over to the side to get that indirect heat to bring it up to the perfect temperature. And as they're sitting there doing all of that, the smell of that comes wafting over the fence into your side of the yard. Of course, with social distancing, you can't go over there and have any of it, but you can smell it. And can just the smell of that coming from outside of your house even be enough to stimulate your digestive system? Absolutely. And so that's really the key with the central nervous system. It can be inside or outside. Heck, there doesn't even have to actually be a stimulus. If you were to close your eyes and really sit there and spend 15 minutes, and again, now that we're all home, stuck at home, quarantined, you're able to do this. You could just sit in your room for 15 minutes and think about the best meal you've ever had or the best dish you've ever had. And if you really sat there and thought about it and imagined what it tastes like and what it looked like and sat there and really just thought about it, just the cognitive thought of it would have the ability to maybe start to get you to produce saliva or even get your tummy a little rumbly. Right? So these, with the central nervous system, the stimuli doesn't have to be in the gastrointestinal tract. It can actually start before that even occurs. And the other key is if we are going to be using our central nervous system, we are not limited to, excuse me, we are not limited to one organ. it has the potential to be able to affect multiple organs. So again, the salivary glands in your mouth can become active, your gastrointestinal tract can your stomach can start getting rumbly, other things can occur as well. So with these long reflexes, not only can it be inside or outside the gastrointestinal tract, it's not local, but the other key is that it can affect multiple organs. All right. Now, as I mentioned, these are going to be your sympathetic and parasympathetic pathways. Let's talk about the nerves, and we'll start easy. What is the parasympathetic pathway to pretty much all of the digestive system? Most of it, not all of it, but most of it.
Vagus nerve?
There you go. Cranial nerve, good old cranial nerve 10. Our vagus nerve, absolutely. Our vagus nerve, as we talked about, is responsible for about 90% of all of our parasympathetic output. And so it is the majority for most of it. As we'll see, the defecation is actually out of the sacral reflexes from the sacral portion of the spinal cord. But it's mostly cranial nerve 10, and then we'll throw some sacral on there as well for defecation. Sympathetic was a little trickier. Anybody remember what we called those sympathetic nerves that go to the organs below the diaphragm? I'll save you the sprained brains. We called them the Splanchnic nerves. The Splanchnic nerves are the ones that come off of the collateral ganglia. And it is those collateral ganglia that pretty much innervate all the organs below the diaphragm. So we have those splanchnic nerves for the sympathetic, and we have the vagus nerve, a little bit of sacral, for our parasympathetic. So those are the pathways we are going to be taking. There's a lot of words on this page. So let's look at a pretty picture that reminds us of these same things. So, all right, I think we got all of that. So I'm gonna have to clear it so that I can do, whoops, not that. Here we go. So again, here is the key to these two reflexes, the short intrinsic reflexes. And again, with that, it is a local stimulus inside of the organ. There are chemoreceptors, osmoreceptors, mechanical receptors in the organ. Our intrinsic nerve plexus, that enteric plexus, makes the decision. Again, the effector is local, either smooth muscle or glands, and we get a local response. But that same stimulus, that same local stimulus, can also be sent to our central nervous system. When it gets sent through the central nervous system, again, our brain, our spinal cord makes decisions on that, and then it's going to send information to, and again, the key to here is we can affect multiple organs. Right. A great example of this is one we'll talk about, but anybody who's had a little kid knows this really well, right? You have that little kid comes running down to the dinner table, sits down, has 3 bites of food, and then they have to get up and run away from the table again. And why are those little kids getting up and running away from the table? What's happened?
Distracted.
True, it could be distraction, especially this. Now, often what ends up happening is they need to defecate. All right, little kids often will sit down, take 2 bites of food, and when they take those 2 bites of food, suddenly they need to relieve themselves. They need to defecate, right? And so they have to go running to the bathroom, right? And we'll talk about the actual mechanism that causes that to occur, but the short version of this is a local stimulus of the stomach sends a long reflex through the central nervous system to other organs like the small intestine and the large intestine, basically to let them know, hey, new food is on the way. So anything that you're working on right now, you got to move it along down the line. Right? Now, you often don't take one bite of food and then have to defecate because you're much bigger and you have a longer digestive tract. Little kids are smaller, and so as that stuff starts moving down the line, it reaches the end much more quickly, and they're much more likely to have to go and defecate as a result of that. So the key with these long reflexes is that it can be a local stimulus, or, like we said, it can be either outside the specific organ or even outside the whole digestive system. Right? You can smell that tri-tip. You can hear the sear. You can see that Carl's Jr. commercial, right? Or you can be thinking about it. All of those are examples of stimuli that are starting outside the digestive system, but still have the ability to have some effect. All righty. So those are our neural controls. And then, of course, not surprisingly, we also have hormonal controls as well. We hinted at this when we talked about the endocrine system. We said we weren't going to bother listing all of the organs that produced hormones, all of the hormones they produced, and all of the effects of those hormones, because we were going to do it during the digestive system. And so that by the same token, there is no sense in listing them all right now as we go organ by organ. And that's what we're going to do for this system. For this system, we're going to follow that cheeseburger on its mystical, magical journey through your alimentary canal. And as we work our way through, we'll talk about the hormones that are produced in that process. However, the important thing to remember is that many of the organs, in fact, many of the cells of the digestive system can produce hormones, and those individual cells that can produce hormones are called enteroendocrine cells. There are close to 12.5, over almost 24 hormones that are produced by the digestive system. We will not talk about all of them. We'll highlight some of the main ones, and we'll be streamlining it, especially because of the interest in time that we have. But as we've talked about, hormones, because these are going to all be circulating hormones, have the ability to coordinate activity of many of the alimentary canal organs, as well as the accessory organs. Right? The accessory organs are not being directly stimulated by the food. Like I said, the food does not pass through the pancreas, but there are going to be signals in the blood either from the absorption of the food or hormones that are going to be released by the alimentary canal that will be able to control the activity of those structures and stimulate their assistance. And again, hormones, because they have many functions, often can influence other systems as well. So digestion can affect heart rate, can affect respiration, can affect lots of other organ systems as well. All righty, questions on that? All righty, excellent. With that, that is our introduction, all of the basic components of it. And so now what we need to do is talk about some of the basic anatomy associated with this as well. This is going to be a big chunk of material that we're going to need to talk about. So I think even though it is a little bit early, this is a good place to take our first break. So what we'll do is take our first break. For anybody who came in a little late, if you have any questions or anything, I'll happily answer those questions. For those of you who came in a little late, I did mention I'm almost done with the grading, but I'm not done with it yet. I will hope to have it done today, tomorrow at the latest, and then I'll get those things posted sometime tomorrow. And I will likely be curving because I know that that was something that was asked as well. So there will be some type of curve going on with these to assist in that. And then, like I said, and I've also solicited for comments and suggestions about the testing process, because as I've always said, I don't want the test to get in the way of you showing me you have the information. But like I said, also, I'm fearful that that may have happened in this process. So anything that you think could improve it, I would be interested in hearing those comments and concerns, either by email or however that way. All right, so let's go ahead and take our first break. My watch says 8:48, so let's come back at 8:58. right before 9 o'clock, and we will pick up from there. So I'm going to go ahead and pause the recording, restart the recording again. Our goal now is going to be to talk about the basic anatomy, or basic histology, really, microscopic anatomy of the alimentary canal. And one of the keys to this, and we have to add a little bit more to our writing here. So maybe I can sneak it in. No, I can't. All right. Again, one of the important things to remember is we are talking about the basic microanatomy of, and I'm just going to put a C for alimentary canal, and this is going to be from the distal esophagus to the anus. The anatomy of the oral cavity, of the pharynx, and of the basically proximal 2/3 or superior 2/3 of the esophagus are specialized. The anus is specialized. And again, that kind of makes some sense because those are the openings to the alimentary canal, and so they need to be protected, they need to be specialized. So basically everything else has this same basic microanatomy to it. And to that, there are 4 main layers. The first one facing the lumen, because remember, of course, the lumen of this is exposed to the outside world, is our mucosa. And again, mucosa is just a fancy way of saying mucous membrane. Right. Oops, let's spell it right. Our mucous membrane. That is, I have to cheat and do that to sneak it in there. This mucous membrane has 3 sublayers to it. So if we were to look inside of it, and let's cheat first, and we'll go with the lighter green for the first layer. We will go medium green for the second layer, and then we are going to go red for our third layer. As we face the lumen, not surprisingly, this is going to be an epithelial tissue. And that epithelial tissue, you need to make that a little bit smaller. That'll work. That epithelial tissue that lines the lumen from basically the distal third of the esophagus to the rectum is, as I'm sure you remember from 430, a simple columnar epithelial tissue. It is simple columnar. Sometimes it will have microvilli. But not necessarily. All of it does. That is something we have talked about and seen before. And as we know, all mucous membranes then have a connective tissue that epithelial tissue sits on. And with all of our mucous membranes, what connective tissue is that again?
Lamina propria.
True, that is its name based on location. Absolutely.
Areola.
Location, there you go. We're going to call it a lamina propria. Absolutely. And you are also correct in that the actual tissue type is an areolar connective tissue. So absolutely, you have an areolar. It's horrible. Connective tissue that, based on its location, we are going to call a lamina propria. Nothing new there. That is definitely something that we have seen before and knew before, so that shouldn't be new. But notice I also said there is a third sublayer. That third sublayer of the mucosa is what is called the mucosa muscularis. And that mucosa muscularis is basically smooth muscle. And this little bit of smooth muscle has the ability to be able to change the shape of the mucous membrane. A change in the shape of the mucous membrane can help to increase its surface area for absorption. It can help to dislodge things that are stuck. So for instance, if you get a little bit of peanut butter stuck on the roof of your mouth, you can use your tongue to kind of dislodge it and move it along the way. So it has that. This little bit of smooth muscle, though, is not what is producing the big, huge contractions that are moving the entire food through the alimentary canal. This is just really changing the shape of the mucous membrane. And this ability, like I said, can affect surface area, it can affect shape, it can help to dislodge things along those lines. So it has this thin layer of smooth muscle, and that's nice because that thin layer of smooth muscle of the mucosa muscularis is going to help us to distinguish our mucosa from our second layer. The second layer, let's go ahead and draw that second layer. And we'll use blue for this. This second layer is what is known as the submucosa.
Quick question.
Yep.
So the mucosa muscularis is not responsible for like peristalsis?
That is absolutely correct. It is not responsible for peristalsis or any of the other gross muscle movements that are used to either propel food or to churn food, right? Because again, we're going to mechanically digest and we are also going to move it from one location to another. Both of those functions of the digestive system are not done by the mucosa muscularis. Not surprisingly, we have a second muscle layer, and that second muscle layer is going to be what is responsible for that. Great question. Any others? All right, excellent. Our second layer is the submucosa. This submucosa is also comprised of areolar connective tissue. However, this areolar connective tissue is not part of the mucous membrane, so this is not a lamina propria. All right, so this is not a lamina propria that we have here. Again, it's important because when we look at this histologically, this right here is going to be areolar connective tissue. This right here is going to be areolar connective tissue. So both of these are areolar connective tissues. There isn't a big difference between them, but there is this little bit of smooth muscle in between. And so the smooth muscle in between distinguishes the mucosa, and therefore the lamina propria, from the submucosa, areolar connective tissue. Now, it's not a lamina propria, but that doesn't mean that there aren't important things in here. There are indeed some important things in here, including large glands and blood vessels. As things are being absorbed from the capillaries and stuff that are here in the mucosa, we go into the large glands and the blood vessels here. But the other important thing that is going to be in here, and I'll draw it as a squiggly line in yellow, is within the submucosa where there are these glands, we also need to control the glands. Remember, we talked about how there is this enteric plexus. And this enteric plexus, and this should also be yellow for consistency's sake. This enteric plexus, this elaborate networks of nerves and that control and give it that intrinsic control. And it actually has two divisions. One of the divisions of the enteric plexus is found here in the submucosa, and that division is what is known as the submucosal. branch, or the submucosal plexus. Let's say it that way. Submucosal. Oops, if I spell it right. The submucosal plexus, and not surprisingly, since here in the submucosa is where there are large glands, it is the submucosal plexus that controls the large glands that are found in this submucosal region. So when we need to produce and release secretions, it is the submucosal plexus that basically controls the glands. Let's just say glands. All right. So again, it isn't so much of a sublayer, but it is this network of nerves is that found within the submucosal layer, along with all of those large blood vessels and the large glands. Our third layer and again, let's make it a little bit larger. Perfect. Is going to be let's go big. The muscularis. Now, this is sometimes called the Muscularis Externa. Oops, if I spell that right. To distinguish it from the Muscularis Mucosa, but you can actually just call it muscularis. So this is the muscularis, whereas this small layer here is the mucosa muscularis, or Muscularis Mucosa, versus the Muscularis Externa that we have up here. And this Muscularis Externa typically has two layers. Those two layers, so let's go ahead and divide it. Both, let's say, start this way. Both layers are smooth muscle layers. The superficial layer, which again, superficial in this case is closest to the lumen, is a circular layer. This circular layer, being circular around the circumference of the lumen, is going to affect the diameter. And for that, I think I need to do it this way. So it is going to affect the diameter of the organ, right? In fact, in some cases, this circular layer enlarges, and when it enlarges, it forms the valve-like structures we call sphincters. And those sphincters, their valves, help to regulate the flow of food through our alimentary canal. Let's go ahead and go consistent. The deepest layer, or the furthest from lumen, is going to be a longitudinal layer. And that longitudinal layer is going to affect the length of the organ. So again, it runs the entire length of the organ. So if the organ, again, if you think of the way muscle works, when muscle contracts, it gets shorter. So if a portion of your small intestine is a foot long and the longitudinal muscle layer contracts, it's going to go down to 9 inches or whatever it is. So you'll get that shortening of the organ in this fashion. Now, here we have our smooth muscle. And of course, we want to control that smooth muscle. So of course, here inside of our Muscularis Externa, again, not just between them, but within the two layers of our smooth muscle, we are going to have, and I'll cheat, let's move this up here, so there, and then I can in between. We are going to have the second division of the enteric plexus, and that is the myenteric plexus. And the myenteric plexus, not surprisingly, controls the smooth muscle. So remember, we basically said that there are two main effects that are going to occur in our digestive system. We are going to change the motility, change the way the muscle contracts, and we are going to change the amount of secretions that are produced with that enteric plexus. So not surprisingly, it has two divisions that do those two things. One division, the myenteric division, or the myenteric plexus, controls the smooth muscle. And the other division, the submucosal plexus, controls the secretions from the glands. All righty. That leaves us with one more layer. And for this, let's go, I guess we'll go brown. This layer is what is known as the serosa, right? Because it is a serous membrane. Let's be consistent. This is the outer anatomy of the stomach. This is the part that is associated with the abdominal pelvic cavity. And as part of the abdominal pelvic cavity, it is, of course, surrounded by our visceral peritoneum, which is another thing we could call it as well, right? It's the serous membrane, or more specifically, it is the visceral peritoneum. And of course, as we know, all serous membranes are comprised of two layers: an epithelial tissue that is on top of a connective tissue. And of course, with a serous membrane, what is the epithelial tissue? You guys should remember this from the respiratory system. It is, of course, our simple squamous epithelial tissue, which is what forms the epithelial tissue. And again, remember this simple squamous epithelial tissue that forms part of a serous membrane based on location. We call, actually, and let's cheat, and we'll write this smaller in a second. And the connective tissue is, of course, areolar. That areolar is not a lamina propria, because remember, a lamina propria is only, an areolar connective tissue that's part of a mucous membrane. But remember that our epithelial tissue, a simple squamous epithelial tissue that is part of a serous membrane based on location, we call it a mesothelium. So our simple squamous does have a name based on its location. It is a mesothelium. And that is the basic anatomy, except when it is not. As we will talk about, and we hinted at this a little bit with the digestive system. If you remember that, I mean, in the respiratory system, normally this comes before the respiratory system. So we get a chance to talk about that in the urinary system. We get to talk about this first. But remember, the kidneys we talked about are in a retroperitoneal position. They are behind the abdominal pelvic cavity. Most of the organs of the digestive system, or let's say it this way, many organs of the alimentary canal are inside the peritoneal cavity. And those that are in the peritoneal cavity are lined on the outer surface by a serosa. Serous membrane, it is the visceral peritoneum that is a simple squamous epithelial tissue on top of an areolar connective tissue. Of course, we call that simple squamous epithelial tissue a mesothelium. Remember also, we produce a thin layer of transudate, well, let's say it this way, serous fluid on the surface. That's what's make these slippery and allow them churn around. And based on location, that thin layer of serous fluid is a transudate. All right, this is definitely stuff we talked about in the cardiovascular system and should have talked a little bit about in our respiratory system. But many is not all. Some of our digestive organs are retroperitoneal, like the kidneys. They are behind the parietal peritoneum, and as such, they do not have a serosa on their outer surface. They don't have a serous membrane. Instead, they have a fibrous, a connective tissue that anchors them to the adjacent tissue. Anchors them in place. Let's say there. Anchors them in place to the surrounding tissue. And in that case, that outer layer is then called, sneak this over here so it's out of the way, move this up here. So our organs of the alimentary canal either have a serosa, or the other thing they are going to have is what we call, so if they, again, anchor them in place, so the retroperitoneal organs have an adventitia as the outer layer. Oops, layer. So it's either a serosa or an adventitia. So you have one or the other. And the determining factor is whether it's intraperitoneal, inside the peritoneal cavity. We refer to that as being intraperitoneal. Whereas if you are retroperitoneal, you have an adventitia. All right? Our pretty simple basic anatomy just got a little bit more confusing, but I still think it does a decent job of describing and illustrating the components here that we need to talk about. I'll go ahead and take a pretty picture of that so we can share that on our Canvas site. But we have a better way of looking at this as well. And that is the pretty picture here from your textbook. Notice as we look at the pretty picture here from your textbook, we see all of those same things that we just finished talking about. Notice starting from the lumen. Here, let me do annotation tools, my highlighter. So notice right here, we have our mucosa, and our mucosa is comprised of 3 layers. We have our simple columnar epithelial tissue and an areolar connective tissue, which based on its location, we call the lamina propria. And then we have a thin layer of smooth muscle known as the Muscularis Mucosa. Notice there can be some small glands. Notice there's even some MALTs, mucosa-associated lymphoid tissues, that will be found within this area. So that is our mucosa. Notice there is more areolar connective tissue outside the Muscularis Mucosa. It has large blood vessels, it has large glands in it, and this is the submucosa. Again, being the submucosa, it is not part of the mucosa, so it is not a lamina propria. As I mentioned, it has large blood vessels. Notice some lymphatic vessels, some glands, all of those big structures. But notice the other thing that it has in here are the nerves that are going to control these blood vessels and control these glands. And that is the mucosal plexus, nerve plexus, part of that enteric plexus we talked about. Here are our two layers of smooth muscle. Notice the two layers are perpendicular to each other. Here, those spindle-shaped smooth muscles are forming the circular layer that goes around the entire circumference of the organ. This controls the diameter of the organ. This is the kind of layer that we saw in our blood vessels. This is the kind of layer that we saw in our bronchial pathways. Right? Those circular muscle layers that basically can control the diameter. In some areas, this enlarges and thickens greatly, and as it enlarges and thickens greatly, it forms an actual valve that can close to regulate the flow, and so this circular layer can enlarge to form our sphincters. However, the majority of our alimentary canal actually has two layers of smooth muscle. One that is circular, and one that also runs the length of the organ. This one that runs the length of the organ is the longitudinal layer. And so when it contracts, of course, the organ gets shorter. Lastly, here we have our serosa, a simple squamous epithelial tissue on top of an areolar connective tissue. Right, so this is our serosa, or again, this would also be our visceral peritoneum. And if you remember, one of the things that we talked about way back in the day in 430 is that when you have a visceral peritoneum that wraps around like our small intestine, it has to connect to the parietal peritoneum that wraps around the entire abdominal pelvic cavity. And if you remember, the way we talked about, and we saw this in our rat dissection, if you remember, is what happens is and I'll cheat and draw this in black. So we have our parietal peritoneum that runs around the outer portion. It comes off the back wall of the abdominal pelvic cavity to come up, wrap around the organ, and then come back down to then continue on its way. And so what we end up with here is a double layer of serous membrane. This double layer of serous membrane, as you can see, is going to be a passageway for nerves and blood vessels and lymphatic vessels and things along those lines. And this specialized structure we call the generic term for them is a mesentery. So the generic term is a mesentery, and as we'll see, each individual mesentery has its own unique name. Now, of course, if this was a retroperitoneal organ, then instead, this outer layer would not be a serosa. Instead, it would be an adventitia. All right. Questions on that?
I've got a quick question.
Yep.
What exactly does the Muscularis Mucosa actually do?
So again, it changes, the Muscularis mucosa is going to change the shape of the mucosa. So it's not going to change the whole diameter, but it could change the shape. It could, as we're going to see, there are going to be folds within the mucosa. And so it could increase or decrease those folds, which would increase and decrease the surface area. That would affect how much absorption could take place. Also, by changing the shape, it can also dislodge any food that may have, or other structures or substances that could attach to the wall of the muscularis. We have this thick, sticky mucus in our stomach. We have that peanut butter that you had for your breakfast, all those types of things. So it can help to dislodge components from it, change the shape of it to increase its surface area, and things along those lines like that. But it is not what is moving big, bunches of food through. It's not what's providing the propulsion. It's not what's providing the chemical digestion. We're just manipulating the mucus, the mucous membrane, and that's basically it. All right, great questions. Any others? All right, excellent. Clear my drawing and take a look at some more pretty pictures. Again, here as we talked about, and again, we just went through this, but we'll go through it one more time. Beat the dead horse since I'm not there to do it in person. Our mucosa has 3 layers to it. The epithelial tissue, which is the simple squamous epithelium. Our lamina propria, the areolar connective tissue. Excuse me. And the Muscularis Mucosa. We talked about the functions of that. Our submucosa is that areolar connective tissue with large glands, large blood vessels, large lymphatic vessels, things like that in it. And also our submucosal plexus that is going to control those glands, control those blood vessels. We have our 2 layers of smooth muscle, the muscularis, circular, closest to the lumen, longitudinal, furthest from the lumen, changing the diameter, changing the length of the organ, and they're controlled by that enteric plexus. Right? And again, as we talked about, with these 2 layers, we are able to produce muscle contractions, and it is the job of this Muscularis Externa, or what we can just call the muscularis, to, among other things, propel food through the alimentary canal. And as we've kind of already talked about before, one of the main ways that substances are moved through a hollow organ is the process of peristalsis. Basically, peristalsis is defined as an alternating rhythmic contraction of the 2 layers of the muscularis. Again, this illustration doesn't do as nice of a job of showing that, but I believe, there we go, I have this pretty one that does. So notice here, for this peristalsis to take place, the first thing that occurs is we get a contraction, a narrowing of the lumen by a contraction of the circular layer. Then we get a shortening of the organ with the contraction of the longitudinal layer. Then our circular layer contracts, then our longitudinal, circular, longitudinal, circular, longitudinal. And by having these alternating contractions of these 2 layers, we are able to, with these rhythmic contractions, be able to propel our food on its mystical, magical journey through the alimentary canal. So peristalsis is the primary method of propulsion. Right? Again, notice primary is the key word there. So there are going to be others. Excuse me, pardon me, but it is the primary form of propulsion. All right, so again, I think the simple picture on the left doesn't do as good of a job of showing this, but I really like this picture on the right that does a really nice job of step by step showing those alternating rhythmic contractions of both layers for that peristalsis. All righty, questions on that? Great question. So the short answer is there are indeed constant, well, let's not say constants. There are indeed contractions that are continually taking place in our alimentary canal. As we talked about, our alimentary canal has some muscle tone in it, so there's going to be some contractions in it that way. There are also going to be, it takes time, 24 to 48 hours, to completely process all the food you eat from a meal. So while you may not be currently eating at this moment, there is still going to be action taking place within your alimentary canal, moving food that you have eaten over the past day or 2. Now, if you were to fast for a couple days, there would, theoretically be no large amount of any kind of food or any substance within your alimentary canal, but there will still be some minor contractions taking place. But absolutely, this peristalsis in particular is stimulated by the presence of food. So when food is present, that is going to be a stimulus for this peristalsis, and peristalsis will be produced. Because think of it this way. Even if you're not eating, you're still producing saliva. And unless you've got a cup to constantly collect it, that saliva is still draining down your throat into your stomach. So, you know, your stomach is still producing acid. So those fluids need to be moved through our alimentary canal as well. So yes, there is going to be constant movement, but obviously it is enhanced when food is present. Great question. Any others? All right, peristalsis is one of the primary forms of movement that take place in our alimentary canal, but it is not the only. There are going to be some specialized movements that can take place in different organs, and a great example of this is the process of segmentation. Segmentation only occurs in the small intestine. And in the small intestine, the big difference between peristalsis and segmentation is alternating contractions of the circular layers, of the circular layer. All right, so notice what's happening here. Here we have that cheeseburger you had for breakfast, and they've highlighted different colors to this to kind of show you the different amounts of food, or as was mentioned appropriately, bolus, that would be found in here. Although really at this point it's chyme, but that's beside the point. The food is in here. And notice here, there are 3 regions of the circular layer of the small intestine that have contracted. So these 3 regions have all contracted. But then what happens is a different region of the small intestine contracts, this one and this one. So notice now these 3 relax, these 2 regions contract. And so now we get a squeezing at these 2 locations. And then squeezing at the 3 original. So again, the way I always demonstrate this, and hell, it's easiest to do this just with my hands. All right. We have these 2 regions, and one contracts and then the other contracts, and one contracts and then the other contracts. We have this alternating contraction of the, oh, do you see? Our ARC has posted digital virtual backgrounds for Zoom, and they had a classroom one. I missed my whiteboard, so I used that one. But what we have is this alternating contraction of just the circular layer. Notice when we do this, the goal of this isn't to, get me off there because me picking my nose can be distracting. All right. Notice when we do this, what we get here is a mixing of the food, right? With this segmentation, there is only minor propulsion that takes place with it. Instead, the function of this is really about mechanical digestion. This is about mixing the food, right? So again, I know every single person in this class, when they take a bite of food, they chew that food the 30 times you're supposed to before you swallow it down. But I know this is shocking, but believe it or not, there will be people who take a huge bite of that cheeseburger for breakfast, take 2 chews, and then swallow it down into their gullet. I know, it's horrifying to think of, but it occurs. So luckily for those people, we can have some churning that occurs in the stomach. We can have this type of mechanical digestion that is taking place in our small intestine as well, where we're basically mashing the food back and forth. So that is definitely one of the advantages of this segmentation. But also in our small intestine is where we are going to be adding a lot of the enzymes. And so this segmentation is going to mix the enzymes into the food, with the food, mixing it back and forth. So that's going to help to enhance chemical digestion. The other thing that this segmentation is going to be doing is basically taking the food and rubbing it back and forth over the surface of the small intestine. And the small intestine is where the majority of the absorption is going to take place. So if you think about it, by taking the food and rubbing it back and forth on the small intestine's wall, it's kind of like increasing the surface area. And that increase in the surface area obviously is going to assist absorption. So notice this segmentation of the small intestine does a lot, but propulsion is only one of the minor influences of it. It's much more involved in the other processes that are taking place in our small intestine. That's probably one of the best examples, but there are plenty of others that we'll talk about more when we get to them. For instance, in our large intestine, we are going to have both haustral contractions and mass movements. Not sure why that's written that way. I don't like that. I don't even know if I can change that. Oh, here, I'm gonna have to cheat. Let's just do this the easy way. Give me 1 second. And that. Bring that. There, I like that better. Come on, share my screen. There we go. Closing everything and I have to open everything back up again. There we go. Excellent. All right. We did that, we did that, we did that, we did that. There we go. In the large intestine, we also have examples like mass movements and haustral contractions, which are processes we'll talk about when we get to the large intestine. So obviously peristalsis, and again, here's the last thing. All of these are produced by the Muscularis Externa. Right, it is the Muscularis Externa that is responsible for all these motions. Like I said, all the Muscularis Mucosa does is control the shape, really, of the mucous membrane. So whether it's a mass movement, haustral contraction, segmentation, churning in the stomach, all of those things are the function. And of course, peristalsis. All of those things are the function of the muscularis. All right. Lastly, as I mentioned, many of the organs of the alimentary canal, like this illustration that they've shown us, are surrounded by our visceral peritoneum, or what we call our serous membrane, or serosa. It is a simple squamous epithelial tissue on top of an areolar connective tissue that connects by this double layer of serous membrane to the parietal layer by that mesentery. And again, mesentery is singular, mesentery is plural. Mesentery, again, and this is important, mesentery is the general term for one of these double layers of serous membrane that anchor an organ in place, or really loosely, because that's really the key to this. They're going to loosely anchor that organ in place, allowing it the flexibility to be able to move and flow. But as we'll see in just a minute, every single specific mesentery has a specific name. So of course, on the exam, I can point at one of these and ask you for the general term, and the general term is always going to be mesentery. But I could also ask you for the specific term or the specific name of it, and then that will be whatever it is. And we'll talk about those and see those in just a minute. But most is not all. There are some organs that are retroperitoneal, and I don't think I wrote that on this. Nope, I didn't, so let's do ahead and write that here. Those that are retroperitoneal, which again is hopefully a term we know now from talking about the kidneys, but if not, we will talk about it in more depth in just a minute. And if it is a retroperitoneal organ, then its outer layer is an adventitia, a fibrous connective tissue whose job is to not loosely, but tightly attach it to and anchor it in place. Clear. All righty. So let's talk about this some more. As I mentioned, our mesenteries are double layers of peritoneum membrane with a little bit of real connective tissue in between. There are access routes for blood vessels, nerves, and lymphatic vessels. Again, their job is to loosely attach the organs, right? It allows things like your stomach and your small intestine to be able to twist and turn and churn to deal with the food inside of them without getting entangled. Right? Many of you may not be aware of this, but in ancient times, earphones had cords on them. So back in ancient times, when you first got a phone, and heck, even before you had phones, when you had your Discman that you were listening to, you had a pair of earbuds that you would put into it, and they had a cord that connected to the device so that you could listen. And one of the amazing things about these earphones is that you could take them with their cord, put them into your pocket, and then instantly reach in there and pull them out, and they would be twisted into a massive knot that would take you a half an hour to untangle. And it happened instantaneously. It was truly a work of magic. All right, now all you have to worry about is losing one of your earbuds and then having to buy new ones. But back in ancient times, we had these cords that would constantly get tangled. And we don't want that to happen to our organs of the alimentary canal. So we want to prevent that entanglement, but give them that loose attachment so that they're able to move freely enough to process the food inside of them. And that is what this mesentery does. Right? But like I said, it doesn't call it for all of the organs. Some of the organs are retroperitoneal. And here we can start seeing some of these examples. Notice here's our liver. This is a nice sagittal view. Our liver, our stomach. This is part of our large intestine, part of our small intestine, right? Things along those lines. Notice they are all inside the abdominal pelvic cavity. The abdominal pelvic cavity is defined by this green line, right? That is the parietal peritoneum. And so this, of course, is the peritoneal cavity. Notice in blue, all of these intraperitoneal organs, and again, that is the key word, intraperitoneal, are, as we talked about, lined by a visceral peritoneum and again on this illustration we see that in blue. So our liver has a visceral peritoneum, our stomach has a visceral peritoneum, this part of our large intestine, these parts of our small intestine. So we see all of those. But what we also see is that again these serous membranes must connect to each other. So notice there's a little bit that comes off the wall to become the visceral around this. And then notice there is a little bit of areolar connective tissue that comes off the liver to help to anchor the stomach in place. And the same thing here as well. This double layer of mesentery, again, all of these general term for all of these is a mesentery. All right, and let's highlight that in a pretty color, red. All right, make it a little bigger. 36. Oh, helps if I spell it correctly. General term is a mesentery. Right? There. And there, that works. Perfect. General term for this is a mesentery, but notice we have specific mesenteries as well. Notice the specific mesentery that anchors the stomach to the liver is called the lesser omentum. Notice from the other end of the stomach, there is a mesentery that comes off of the stomach, drapes down like an apron over a majority of the abdominal pelvic cavity, and then comes back up to the large intestine. And this one here is known as the greater omentum. So notice the stomach has a lesser omentum and a greater omentum. We haven't talked too much about the anatomy of the large intestine yet, but the large intestine helps to form a frame in our abdominal pelvic cavity. And this is the perpendicular part of it known as the transverse colon. And this transverse colon, notice, is anchored to the back wall by a mesentery known as the transverse mesocolon. The small intestine, the majority of the small intestine, not all, but the majority of the small intestine is anchored in place by a mesentery. And notice it says mesentery. Mesentery, remember, though, is our general term. So to distinguish it, what we are going to do for the mesentery that holds our small intestines in place, we are going to refer to it as the term mesentery proper. Oops, if I spell it correctly. So the mesentery proper is the specific name for our small intestine mesentery. The generic term is still going to be mesentery. So we see that nicely. The other thing we see nicely in this is that, notice things like the pancreas, things like the first part of the small intestine known as the duodenum or duodenum, are behind the parietal peritoneum. Remember the kidney, let's put a big K here to remind us of that as well. That's not a K, there we go. That kidney, remember we saw, was back here behind the parietal peritoneum. So behind the parietal peritoneum, and so those are what we call those retroperitoneal organs. All right, let's change the view and take a look at it from a different standpoint. Clear my drawings. I love this picture because remember what we see here is that greater omentum. Remember that greater omentum, as we talked about, is a drape that kind of drapes over the front of the abdominal pelvic cavity. So what they've done here is they've lifted it up out of the way. So it was coming off the stomach, which we can't see anymore. It was coming off of that, draped it down from there. And here we see the transverse colon where it attaches to the front. And so here on the back is where a transverse colon attaches to the posterior wall of the abdominal pelvic cavity. So this is our transverse mesocolon. Whereas here we have that mesentery, and again, we're going to call it the mesentery proper. We're going to give it that specific name. Notice there's one more mesentery here that you are responsible for. Again, we have not done the anatomy of the large intestine yet, but let's do this quickly. Portions of it, it frames the abdominal pelvic cavity. We have the ascending colon, the transverse colon, the descending colon. And notice we have this S-shaped region, and this S-shaped region is called the sigmoid colon. Well, the sigmoid colon happens to be held in place by the sigmoid mesocolon. So those are all of the mesenteries. I believe those are all the mesenteries you're responsible for. Hopefully you have your anatomy list in front of you. You are looking at these while we're doing that. If not, first and foremost, shame on you. But secondly, make sure you have that list so as we go through these things, you can be responsible for them. I was looking at the materials that are available to us on that CRC site. There are many, but not all. There are some good things we actually have in the classroom that I believe some of the other instructors have pictures of, models, and materials. If I can get those, I will provide those for you. And then once I provide those for you, those become fair game on the exam. But I will make them available for you. And we're also in the process of working on our own site, but I'll be honest, it probably won't be done in time to help you. I will provide you with whatever pictures I can, and then anything that I provide you for is fair game on the exams. But those are the specific mesenteries that you are responsible for. Here, notice that greater omentum has been cut. It's that big drape that comes down. And here, by lifting up the liver, we can see that lesser omentum that anchors the liver to the stomach, and then the greater omentum. Again, the same way we haven't talked about the anatomy of the large intestine, we haven't talked about the anatomy of the stomach yet either, but I will cheat. Oh, and for this, I'm going to use the highlighter, and I'm going to use purple. No, brown. This region of the stomach is called the lesser curvature. And notice the lesser curvature is where the lesser omentum connects. This outer region here is the greater curvature. And notice, conveniently enough, the greater curvature is where the greater omentum, which again here has been cut, but where that greater omentum would be located and comes down. So that's very convenient. The lesser omentum connects to the lesser curvature of the stomach. The greater omentum comes off of the greater curvature. All righty. Questions on that? All right. Now, as I mentioned, the peritoneum does not cover every organ in the digestive system. Some of those organs are either intraperitoneal or, as I said, retroperitoneal. Here we have the pretty picture from the textbook that kind of shows this difference between intraperitoneal and retroperitoneal. Here's an organ like the small intestine. Here's the organ like the stomach and the liver that are anchored in place. And notice they do distinguish a dorsal mesentery from a ventral mesentery. Those aren't terms we're going to worry about distinguishing. We just need to know the generic mesentery and then obviously the names of the specific ones that are going to be located there. But notice there are many organs that actually start in the abdominal pelvic cavity and are brought back posterior behind the, oh, do I have room? Maybe, maybe not, I'll cheat. It is brought back behind the parietal peritoneum, anchored in the wall. And again, these will have an adventitia that wraps around them. So then the question becomes, what organs are found in this retroperitoneal position? So let's do this. Let's go ahead and, oh, does that work? Yeah, that's what I want. I want that there. Perfect. And then, how big is that? Too big. There. Excellent. So as we work our way down the digestive system, when we talk about organs that are retroperitoneal, if we're thinking about the digestive system, the first of those is the esophagus. The esophagus is anchored into the posterior wall of the abdominal pelvic cavity. However, the stomach, where it connects to, is not. From the stomach, food goes into the small intestine. And as I said, most of the small intestine is found in the abdominal pelvic cavity. However, the first part of the small intestine, which is called the duodenum or the duodenum, is not. It is actually anchored to the posterior wall. So notice the stomach is intraperitoneal, but the esophagus that feeds into it and the duodenum that feed out of it are both retroperitoneal. They actually help to anchor that stomach in place, but still allow the stomach to churn, to produce all of the mechanical activities that it is able to and needs to be able to do. From the duodenum, it feeds into the jejunum and the ileum, the rest of the small intestine, which then feeds into the large intestine. The large intestine, if you remember our rat, has a big, huge pouch-like structure called the cecum, and that cecum is intraperitoneal. But remember, we saw the transverse mesocolon, because the transverse colon is in the peritoneal cavity. But as it turns out, the ascending and descending colon the ascending and descending colons are not. So our ascending and descending colons, again, are helped to be anchored in place. within the abdominal pelvic cavity. So they are anchored in retroperitoneal, but the sigmoid colon, the transverse colon are not. All right. So when we think of our alimentary canal, we have those structures that way. As we already learned, because we just finished talking about it in the urinary system, we of course know that the kidneys are retroperitoneal. And even though we didn't talk about them too much in the urinary system, we did talk about them in the system before that. What sits smack dab on top of the kidneys?
Adrenal gland.
There you go. The adrenal gland, or what is also known, because it is on top of the kidneys, if you have trouble remembering that, is also referred to as the suprarenal gland, right? That was another name for it. But adrenal is what most people remember because it produces that adrenaline. But either of those are the acceptable term for that. Notice also, if, not surprisingly, if the kidneys are retroperitoneal and the adrenal glands are retroperitoneal, remember coming out of the kidneys are the ureters. They are also retroperitoneal, but notice the bladder isn't. If we went back to the previous picture, one of the things that we would see, I think we can probably sneak that in. Let's see if we can do that. Maybe a little messy because I've got the writing on it, but I like this picture. There you go. Notice here, we see the bladder is inferior to the abdominal pelvic cavity. So it is not considered to be retroperitoneal because it's actually underneath. And in fact, even though they've got this drawn as green, the peritoneum on the top of it, if you remember, is known as a visceral peritoneum. It has a serosa on its outer surface. All right. So the ureters are retroperitoneal, even if the bladder and the urethra are not. Now, there are also some accessory organs of the digestive system as well, and those include the pancreas. The pancreas, as we also saw from that previous picture, but notice not all of it. It turns out just the pancreas is a feather-like structure that has a head, a body, and a tail. The head and body of the pancreas are anchored into the abdominal pelvic cavity's wall, retroperitoneal, but the tail dangles out a little bit. Now, why? I have no idea. The food's not passing through the pancreas tail doesn't need to contract or move or change shape to do anything. So why the whole thing isn't retroperitoneal is beyond me. But for whatever reason, just the body and the head is. Notice when we get back to the colon, we know the ascending and descending are retroperitoneal. The transverse and sigmoid are intraperitoneal. But that sigmoid colon then feeds into the rectum. And that rectum is actually retroperitoneal as well. Lastly, the other structures, and again, if you think about it, these are structures that have smooth muscle on them, but we don't necessarily want them wiggling around all over the place. And those are going to be the abdominal, aorta, and the inferior, vena cava. So those are the structures. Again, we don't want those moving around or jiggling around in place inside of the abdominal pelvic cavity. We want them anchored. So if you're having difficulty and need some help remembering what the retroperitoneal organs are, they're retroperitoneal, they don't get to play in the abdominal pelvic cavity, and because of that, they are a bunch of sad puckers. And I do mean pucker. So if you are retroperitoneal, you are a sad pucker. All right, so there's your mnemonic to help you remember all of the retroperitoneal organs. All righty, questions on that? Excellent. All righty, I will go ahead and clear my drawings and see what we're supposed to talk about next. Perfect, excellent. With that, we have talked about the basic anatomy, the basic functioning, the basic processing. But as I mentioned, to really understand the digestive system, we need to take that mystical, magical journey that the cheeseburger does through our alimentary canal and see all of the changes that take place to that cheeseburger as it goes on that mystical and magical journey. And so that is what we're going to do. We are going to start our discussion. Normally, we do all of the anatomy first and then all of the physiology. But I think it's much more diverse in the digestive system. So I think it makes more sense to do it regionally, unit by unit by unit, working our way through the system, look at the anatomy, look at the physiology of each step along the pathway. And that is what we're going to do. We will start with our discussion of the mouth and the oral cavity, but then we'll begin our magical mystical journey. And we will begin that after the break because I've been talking for a little over an hour. So now this is a good break point, another good stopping point. So what I'm going to do is I'll act quickly for questions. And then if there are no questions, then we will come back. We'll say 10:15. And then again, at that time, I will restart the recording. But before we take our break, any questions before we step out into our break? All right, excellent. So then with that, I will pause the recording and meet you back here in 10 minutes. All right, any questions before we get rolling on this? All righty, then let's work our way, like I said, into the first step in our anatomical pathway on our mystical magical journey through the alimentary canal, and that is the mouth. And of course, by mouth, we mean the oral or buccal cavity. The oral or buccal cavity is, of course, bounded primarily by muscles. So if we think about it on the lateral aspect, what are some of the muscles that help to oh, that is way big. Let's bring this down. What are some of the muscles that are responsible for bounding the oral cavity?
Orbicularis oris.
Excellent. So the anterior border is absolutely the orbicularis oris, which again is a circular muscle. So it acts as that valve that we talked about for regulating movement into and out of the oral cavity. Of course, being called the buccal cavity. Why is it called the buccal cavity? Well, because your cheeks are the buccal region of the body, but also why might it be called the buccal region?
Buccinator muscle.
There you go. Buccinator muscles, right? That is that blue steel muscle that allows you to, again, squeeze your cheeks. You could argue the masseter, temporalis. Well, masseter definitely, not the temporalis, but maybe the masseter helps to form that boundary. So we have those that way. We have the lips on the front. All right. Posteriorly, it is, of course, continuous with our oropharynx. All right, so the good news is some of this anatomy is going to be familiar to us because we just finished talking about it with the respiratory system. So we have that oropharynx, which is the posterior part. There are the two arches that are located there as well as where's the fosse in between. And maybe we can look at that anatomy again because some people struggled with that on the lab exam. Okay. Superiorly, what's the superior border?
The hard palate.
There you go, absolutely. It would be the hard palate, which of course is a skeletal feature, or really skull feature. And of course, like all skull features, it is comprised of two or more bones and bone features. And so I'll give you guys some help here. You, of course, remember that there are 4 bones that play a role in forming our hard palate. They are the left and the right maxillary bones, or maxilla, and let's actually do it this way. The left and the right palatine bones. And of course, not only do we need to know the bones, but we need to know the bone features. For the maxillary bones, it is the palatine process. So the palatine process of the left maxilla, palatine process of the right maxilla. And of course, that means that they must be the maxillary processes of the palatine bone. No, of course not. Anatomists hate us. So it is the horizontal processes, or remember, plates of the left and right palatine bones. And if you remember, we have, and I'll do a horrible job of drawing this. If you looked at the inferior part of the skull, the hard palate has this kind of a look to it, where it has 4 sutures that hold these 4 bones together. So bone 1, bone 2, bone 3, and bone 4. And of course, if this was an inferior view looking up, bone 1 would be the right maxilla, and it would be the palatine process of the right maxilla. 2 would be the palatine process of the left maxilla. 3 would be the horizontal process of the right palatine, and 4 would be the horizontal process of the left palatine. So there are 4 bones and bone features that form the roof. And what forms the base? What forms the floor of the oral cavity?
The tongue?
Yep, probably the tongue is probably a good answer to that. I mean, I guess the tongue does elevate, so we do have the underside of the tongue, so maybe you could say the mandible, really, but it's really the connective tissue in the mandible that is able to do that. So I think tongue is probably a good answer for that, for those boundaries of it that way. Let's move forward. Oh, so now I gotta erase all that. The oral cavity, because it needs to withstand a lot of abrasion, that hot coffee you had for breakfast, that sharp tortilla chip you had with your dinner, right? The acidity of the wine that we're all constantly drinking. So of course, what tissue type lines the oral cavity?
Non-keratinized.
Yep, non-keratinized. What's the rest of it?
Stratified squamous.
Stratified squamous, excellent. Epithelial tissue. And of course, it's a mucous membrane, so it sits on top of an areolar. Connective tissue, which of course, based on its location, is a lamina propria. Absolutely. Now, again, we're in quarantine, so only do this with people that you are being quarantined together with. Don't find some random person on the street or the DoorDash person that comes through your door. But if you have the opportunity to look inside of the oral cavity of another individual, one of the things that you may see around the teeth, on the gums, maybe the roof of the mouth, and sometimes on the tongue, you may see a little bit of whitishness to it. That whitishness that you see there, and again, I'm not talking about the saliva, that will be in there as well. But abrasion to the non-keratinized stratified squamous epithelial tissue of your oral cavity can actually cause some of those cells to keratinize slightly. So that whitish bit to the, like I said, the roof of the mouth in some areas, or sometimes in the gums, sometimes on your lips, especially if you have braces or things along those lines, you can get a little bit of whitishness in there. So you can get some minor keratinization that can actually occur from abrasion and abuse inside of your oral cavity. And there you go. So the gums, the hard palate, the dorsum of the tongue can all be slightly keratinized in places because of that abrasion that takes place. Right, kind of the same way we talked about calluses forming on your hands. It's kind of like an oral callus. So again, those kind of things can occur. As we mentioned, we have the cheeks, which are the primarily buccal muscles, but you could argue the masseter is lateral to that. The lips, or more specifically labia, and as you can see here, we have 2 labia. the superior and inferior. And by labia, we mean not just this red part. This red part is a region that is actually called the red margin. What we mean by labia are the folds of skin, the movable folds of skin. So if you think about what is being elevated here is more than just the red margin, but right halfway up to your nose or halfway down to your chin, that is the labia. Another good way to think of this is notice inside the labia, we have a little bit of space between the lip, the labia, and the teeth and gums. This space is the entryway, and in a term that should be familiar from the respiratory system, this area is known as the vestibule, right? So, and again, especially with this picture here, if you think back to middle school, when you were playing Pop Warner football, right, and you would get in trouble, at least this is what would happen in our team. Again, remember, I grew up back east. When you would get in trouble, what our coach would do is he would make you take some chewing tobacco and put it into your lower vestibule, and then make you run laps. Because if you're running laps with chewing tobacco, you have to spit the juice out. You can't swallow your saliva, so it was very dehydrating, and it was a miserable thing that he would do. But when he was pissed at us, that is what he would do. Again, back east. But so we have that labia, that vestibule between the labia and the gums, the gingiva, that region there. The transition skin from the outer surface of your face to the inner surface of your oral cavity is this area known as the red margin. And this is what typically people think of when they think of their lips, right? When you put your lipstick on, most of you aren't putting it halfway up to your nose, right? You're just putting it on the edges, that red margin, that transition area. It is called the red margin because it is a very thin, sensitive tissue with the blood, and because it is a thin epithelial tissue, the blood vessels are close to the surface, and because they're close to the surface, we can see the blood more clearly. We, of course, try to enhance that coloration by putting lipstick on it or things along those lines, stains and other things to it. However, there is a huge advantage to having these close blood vessels. Because the blood vessels are close, oxygenation levels, jaundice, other conditions of the blood can sometimes be seen more noticeably within that red margin area. So when someone gets really, really cold and their lips get blue as a result of that constriction of blood vessels and decreased blood flow to that area and things along those lines. So again, that is that red margin. One of the other characteristics, obviously, as we know, the outer surface of our skin has sudoriferous glands, sebaceous glands to produce sweat and sebum to help to maintain moisture and elasticity of your skin. Our mucous membrane inside our mouth produces mucus to maintain it, but our red margin doesn't have any glands. So then how do we maintain the integrity of our red margin? I know you know. Half of you did it as soon as I asked. What'd you do?
Licking your lips.
Saliva.
Yeah, there you go. You licked your lips. Absolutely. Right. Some of you put on chapstick. I know that happens too. But no, again, remember that's a man-made artifice. We lick our lips, and that's how we maintain the integrity of our red margin that way. As I mentioned, the vestibule is that space between the cheeks and the labia and the teeth. Notice there are two anchor points to this. So the superior labia have a superior labial frenulum. A frenulum, and let's go ahead and it's already written here, so I'll highlight the term here. Let's use a more fun highlighting color, red. That frenulum is a fold in the mucous membrane that helps to stabilize so that your lips don't flap around too much. So your superior labial frenulum is in place. We have an inferior labial frenulum, and notice we have a third frenulum as well. We'll talk about this one more in just a minute, but it is a lingual frenulum. That lingual frenulum, of course, is what anchors the tongue anteriorly. So notice we have not one, not two, but three frenulums. Two that anchor the labia, and one that anchors the tongue. So, of course, you do need to distinguish them by name. Superior labial frenulum, inferior labial frenulum, and lingual frenulum. Like I said, oral cavity proper. We have here the hard palate that forms that boundary between the oral cavity and the nasal cavity. As we talked about in the skeletal system, sometimes that joint between the two maxillary and the two palatine bones doesn't fully form, forming that cleft palate. And cleft palate can also lead to a cleft lip. So, it affects, your physical characteristics, but more importantly, it can influence your ability for the baby to be able to suckle, because if the oral cavity is open to the nasal cavity, then when they're trying to latch on to the breast and suckle, air can get in through the nasal cavity and they can't get a proper seal. Notice there is a bit, and I like this picture a lot, it does a really nice job of showing this, and I want to make sure we emphasize these things. Pay attention to this one, because these are some of the things that we needed to know for the last test, and a lot of people were not clear about. Notice, and again, your chart does a great job of showing this, but this picture does a pretty nice job of showing it as well. Notice there are two main arches that are located within the oral cavity. One goes from the roof of the mouth down towards the tongue, and one goes from the roof of the mouth down towards the throat. So the one that goes from the roof of the mouth towards the tongue, not surprisingly, is the palatoglossal arch. The one that goes from the roof of the mouth towards the throat is the palatopharyngeal arch. Notice, if you have two archways, you have an indentation in between. And this indentation in between is that region known as the fosse. Or fosse is plural. I don't see, and let's change the color of that to red to make that stand out more, and give myself a red arrow to point this out. These spaces are the fosses. And notice the spaces aren't completely empty. In the fosse is where you will find the palatine tonsil. So again, those are some structures that people struggled with on the lab exam, and you're going to get a second chance at it on this exam. So make sure you're comfortable with those four structures. We have the two arches. We also have the space in between the fosse and then the palatine tonsils that are located in the lateral aspects of that. So we have those there. Oops, and okay, oops. And so I think that is it for the anatomy. We'll talk more about the tongue in just a bit, but I think we talked about all the muscles and everything else that are associated with that. All right, excellent. Now let's talk about the functions of the mouth. Remember, as we talked about, there are 6 basic functions of our digestive system, really 6.5, 7 being secretions. And if you think about them, almost all of them happen in the mouth, right? Certainly there is going to be ingestion, right? Some would argue that this is really the only voluntary act. In the process, deciding whether or not you want to shove food into your mouth. And again, I know that's something we're all struggling with, that we're now being forced to be at home with our refrigerators all of the time. However, as we'll see, that's not actually true. There are a couple other voluntary acts. Obviously, while shoving food into your gullet is a voluntary, at least it seems like voluntary, unless you're watching TV, then it's just subconscious. But swallowing is a voluntary act, and thankfully also voiding of our feces, defecation, is a voluntary act as well. So again, while obviously it is one, so maybe that's what we should say instead. One of the voluntary acts that we have, that's shoving the food. And again, the key here is in the mouth is the only location where this occurs. Obviously, if someone has a feeding tube, right, or if they've had stomach cancer or something like that, so they've got, a feeding tube going straight into their small intestine or something like that along those ways, that is a way to get food into our alimentary canal, but obviously that would not be considered ingestion. Again, as we talked about, using our accessory organs, the teeth, and the tongue, we are responsible for mechanical digestion, that chewing, or again, more appropriate term, the mastication of our food that is going to take place. And like I said, as I'm sure you all know and you all participate in, you should masticate each bite of food that you take 30 times before you swallow it down. And I'm sure everybody does that. Those are obvious ones. As we also talked about, we have that 6.5, we have that secretion. And obviously the oral cavity is one of those locations where we are producing a secretion. That secretion is saliva. But remember, we didn't give secretion its own function, because basically the things that are secreted are going to serve one of two, or sometimes both functions. They are going to play a role, or they're going to serve a role in some other function. Right? In the case of saliva, there are a couple of functions it helps with. Saliva helps in the formation of, as a term that was already used by one of our students, a bolus, right? I take that nice, beautiful cheeseburger, I put it on the plate, right? I take a bite out of it, I chew it to 30 times, and then I spit it back on the plate, right? At that point, nobody wants it anymore. They would have been perfectly happy with the cheeseburger. I haven't done much to the cheeseburger other than break it down into parts, but in that process of mastication, I have also mixed it with saliva. And as I've mixed it with saliva, I've formed a substance that we now call bolus. One of the things that we're going to talk about in terms of food, and let's write this up here in a different color so it's just something we have in mind that we're thinking about. And again, this is not a novel concept that you don't know. Food undergoes changes on its journey. Right? Obviously, it comes out of you looking different than it went into you. But like I said, even if I just put it in my mouth and chew it and spit it out, it looks different as well. If I swallow it, let it sit there for an hour, and then bring it back up, it looks differently. So food undergoes changes or has different iterations. That we are going to refer to on its mystical, magical journey. And that first iteration, the first change, occurs in the oral cavity, where we have the mastication, where we have the mixing with saliva. And so when we chew it and mix it with saliva, we form our bolus. So that saliva, and the reason we form that bolus is we form that bolus because when we form that bolus assists in propulsion. By mixing it with the saliva, we basically add mucus to it, grease the wheels, make it more slippery, make it easier for it to be swallowed. All right? So that is definitely one of the functions of saliva. The other function of saliva, and again, digestive function of saliva, is it contains enzymes. In particular, it contains an enzyme called salivary amylase. And being an amylase, it is going to help in the breakdown of what macromolecule?
Carbohydrates.
Carbohydrates. Excellent. This is a fun experiment that you can do for yourself now. Because again, as I'm sure you know, like me, right, because you can't get to the score as often and everybody's now hoarding yeast, bread baking has become a second hobby for pretty much everyone. So what you need to do is go grab a cube of one of those bagels or buns or whatever it is that you've baked. Take a little bit of that white bread, cut it, take a paper towel to dry off the surface of your tongue, and put that cube right on the surface of your tongue and let it sit there. What'll happen is, as you start to produce saliva, that saliva will mix with the bread, and as the salivary amylase starts to break down the carbohydrates, you will actually taste sweetness. You will actually taste the sugars on the surface of your tongue as it breaks it down. So we have that, again, chemical digestion that is taking place there thanks to the saliva as well. And then, of course, that saliva also provides protection. Oops, where did that go? We know the immunoglobulin A antibodies are located in there. The lysozymes are in there to have that antibacterial component. So again, we have all of that protection that is occurring from that saliva as well. And those are, of course, non-digestive functions. So again, that's why secretion kind of is its own function, but really anything that is secreted is serving some other function, like propulsion, like digestion, either chemical or mechanical or something along those lines. All right. Notice another enzyme is produced in our oral cavity, and it is a lingual lipase. As the name would indicate, it is produced by the tongue. It is produced by the tongue, and it is a lipase. So while our salivary amylase, as we talked about, is going to digest carbohydrates, and again, obviously that starts in the mouth. Our lingual lipases play a role in the digestion of fats. Does that mean we could do the same thing? All right, go to that nice big pot roast that you have, cut off a little cube of fat, put it on your tongue, and as you put it on your tongue, your lingual lipase will mix with it, and you'll start to be able to taste the acidity of the fatty acids or the alcohols from the breakdown of the glycerol or anything like that while that occurs?
No.
No, of course not. And that's because it is actually produced in an inactive form. Its job is to start the breakdown of fats, but it isn't effective until it reaches the stomach, which ironically is when the salivary amylase stops working. And of course, what is the reason for that change?
It needs the acid from the stomach.
There you go, pH difference. The pH in your mouth is around 7, whereas the pH in your stomach is about 1 to 2. So what happens is that lingual lipase is produced in an inactive form. As we know, proteins are sensitive to pH changes. So when it goes down into your stomach and the pH becomes acidic, that lingual lipase changes shape and actually becomes functional. So while it's produced in the tongue, mixed with the food in the oral cavity, it does not actually start to break down our lipids until we get to the stomach. And that's going to be an important thing too. One of the guaranteed essay questions you will have on this exam is we are going to talk about the entire pathway of which our major macromolecules are going to be absorbed. We are going to need to identify all the locations where they're broken down chemically and mechanically, all of the enzymes responsible for that, all of the processes responsible for that. Then, once they get to the small intestine, we are going to be responsible for identifying the pathways of how it crosses both the apical and the basal surface. And we need to make that distinction. If you think about it, this is similar to the essay question we had with the urinary system. In the urinary system, we had reabsorption of amino acids, of glucose, of bicarbonate, and sodium. And many of you talked about it, but you need to make sure you're distinguishing what happens on the apical surface from what happens on the basal surface. And so it's going to be a big, huge, elaborate process we'll get to either later this week or maybe not even till next week, right before the exam. So that's going to be fun. But you have the material to look at to be able to prepare for that. But I guarantee that'll be a question. And so obviously notice when we're talking about chemical digestion, we can see that chemical digestion of carbohydrates starts in the oral cavity with the salivary amylase, whereas chemical digestion of lipids starts in the stomach with the lingual lipase. All right, so we have mastication for mechanical digestion, chemical digestion, which can start here. Obviously, defecation does not take place in the oral cavity, but absorption actually does. There are certain simple molecules that can be absorbed in the oral cavity. Water can be absorbed in the cavity. Some medicines, alcohol. These are all some of the things that can be absorbed. And again, you may not have thought of it in these terms, but you're aware of it. Because when grandpa's having one of his episodes, right, what are you supposed to do? You take that nitroglycerin pill and you put it underneath his tongue, right, so that he's able to absorb it that way. There are certain medications or mineral drops, not that I'm accusing you, I'm encouraging you to take mineral drops or things along those lines, but some vitamin drops and mineral drops are things that people will drop under their tongue for better absorption and things along those lines as well. So absolutely, it is limited, right? And that is the key. It is definitely limited. It is not the primary place where absorption takes place, but there is definitely some absorption that occurs in the oral cavity. All righty. Questions on that? All right, let's go ahead and clear that. Switch to this. And then, of course, obviously, from a digestive adjacent process, obviously, we have our sensory input in here, and that is, of course, our sense of taste. While taste is not required for digestion, it definitely helps to stimulate or suppress the process. So it is definitely related to, can affect, let's say it that way, digestive processes. So it is digestive adjacent, let's say. All righty. So that is the oral cavity and its functions. While we're here, we do need to talk a little bit about the accessory structures. One of those accessory structures is, of course, the tongue, as we have talked about. Its function from a digestive standpoint is twofold. Its job is to present the food to the teeth for mastication to occur, and also to present food to, and again, really at this point, it should be the bolus, because you've masticated it 30 times. You need present the bolus to the oropharynx for swallowing. Of course, from a non-digestive standpoint, its job is to produce speech, right, and things along those lines as well. I'm sure there are other uses you can think of for a tongue. If you were to look at the surface of the tongue, you can see that it is not smooth and uniform. There are actually 4 major types of bumps, raised bumps, or what we more appropriately call papillae, along the surface of them. The largest of these are what are known as the circumvallate papillae. Although I will be honest, more and more people are getting tired of writing circumvallate out. So instead, you will more and more commonly just see, vallate papillae, and I will accept both circumvallate and vallate papillae. These are the largest papillae, and let's go ahead and use my highlighter for this, and we'll use blue to indicate this. They form kind of this large, prominent V-like structure at the dividing point of the tongue. Our tongue is kind of divided up into 2 parts. We have the anterior 2/3. This is the part that you more freely move about in your mouth, use for speech, stick out at the person next to you, do all of those types of things. Whereas the posterior third is what is covered with those lingual tonsils, really more of a lymphatic structure than it is a digestive structure. It doesn't have the mobility that the rest of the tongue has. It is anchored in place and is covered with those lingual tonsils, those lymphatic follicles that we talked about. And so our circumvallate papillae kind of form a boundary between them, although you will notice there is a small minor indentation between the circumvallate papillae and the lingual tonsils, and that is actually known as the terminal sulcus. The sulcus, remember, of course, is a groove, so that we see that there. The second largest, and I'll go ahead and continue to use my highlighter for this, but let's change the color to brown now. The second largest are going to be the fungiform papillae. Fungiform because they are kind of round, mushroom-shaped cap-like structures, and these are these prominent bumps that we see all over the surface of the tongue. Both these, the fungiform and the circumvallate papillae, both of these contain taste buds. Taste buds are the specialized sensory structures that are used to perceive our sense of taste. So fungiform papillae over the entire surface, these circumvallate near the posterior part of the tongue, all of these are what provide us with our taste sensations. Notice though, there are two other types of papillae. The first are the foliate. Foliate comes from the same root of the word foliage because it has kind of a ruffled border, like the ruffled border of a leaf. And we see that, and let's go ahead and stick with my highlighter, but change colors again to green this time. Over here on the lateral aspect of the tongue. So as you look at the lateral aspect of the tongue near the back, you'll see it has this kind of ruffled edge to it. One of the interesting things about these foliate papillae is in adolescents and infants, it contains taste buds. These taste buds are primarily sensitive to milk sugars and milk fats. And of course, why might it be convenient to have an infant have taste buds that are specialized for milk sugars and milk fats?
So they enjoy the taste of breast milk.
Exactly, because that's what that's their primary primary source of nutrition, and we want them to like drinking breast milk, absolutely. However, one of the interesting things, well, there's two interesting things about these. The first is that as we age, we lose our taste buds on these foliate papillae. So as adults, none of us have them there anymore. And there is speculation that the presence of these foliate taste buds may be what tends to make some adolescents more finicky to what it is that they eat. Right now, again, a mushroom you barely taste. You can barely taste broccoli, right? Those are things, or tomato, or things that don't really bother you, right? But many adolescents would rather have their left arm ripped off than have to eat a mushroom. And one of the reasons that is speculated is they may be more hypersensitive to some of those flavors that we have dulled to or become more accustomed. Some of it is habituation, right? Some of it is the more you eat something, the more familiar you get with it, the more comfortable you have with it. But, right, there are also some kids that will eat nothing but mac and cheese for the first 3 years of their life, right? Because they hate the taste of broccoli and they hate the taste of all sorts of other things. And one of the reasons for that may be this heightened sense of taste that they get from these foliate papillae as adolescents, which is also why most of us grown out of it. So while there's plenty of little kids who will eat nothing but mac and cheese, there aren't too many 35-year-olds that do. All right, now that still leaves a tremendous amount of the tongue. The rest of this tongue that looks smooth and pink, and I guess we'll see if the yellow highlight shows up, like this part here and this part in here. It looks all smooth and uniform, but as we'll see when we look at it under the microscope, it is not. Instead, there are these tall, narrow, sharp papillae that stick up. These tall, sharp papillae that stick up are the filiform papillae. So the rest of the surface of the tongue is covered by the filiform papillae. These filiform papillae don't and never have contained taste buds. Instead, the filiform give us basically tactile sensation. Right? If you were to, for instance, take a marble and assuming it was clean, put it on the surface of your tongue, right? You obviously there would be nothing to taste, but you'd be aware it was there. And so that's because of the tactile sensation that comes from it, right? If I warmed it in the oven before I put it in your mouth, not to the point where it burned you, but or I put it in the fridge for an hour before I put it in your mouth, you'd have that temperature sensation. Right? As well as if I did put it in the oven for an hour and then put it in there, pain. Those types of sensations all come from the filiform papillae. So those filiform papillae over the surface do, while they don't provide us with a sense of taste, they do provide us with other tactile sensations. And the other thing they produce for us is friction, right? This is normally the point where I would ask people how many people here have a cat or have seen a cat before, and you guys would raise your hands telling me how many of you have done that. But if you've ever seen a cat drink water, basically they don't put their face into the water. What they do is they use their tongue. They put their tongue into the water, and their tongue have very pronounced filiform papillae, all right? By a quick show of hands, and you guys can actually say yes or raise your hand. How many people here have ever been licked by a cat before? One person. Excellent. The rest of you have to get out more often. Oh, there's a couple more. Excellent. All right, perfect. Yeah, a fair number of you have either been licked by or maybe seen someone licked by. And again, it has a very rough texture to its tongue. It has very pronounced filiform papillae. And the reason for that not only is because it allows it to use that friction to capture water when it is drinking, but more importantly, that cat, it is a predator. And typically the things that it eats aren't things that want to be eaten. Now, I know in fairness, your cat, gets stuff straight out of the can or whatever else that it is that you do for them. But in their natural environment, they are predators. And the things they eat don't want to be eaten. And so having that friction on its tongue helps it, right? Us being omnivores, our tongues are not nearly, our filiform papillae are not nearly as pronounced, which is why being licked by your boyfriend and being licked by a cat is a different experience, right? Hopefully it's not nearly as rough of a surface from it because our filiform papillae are not nearly as pronounced. All righty, I'm gonna go ahead and erase that. Of course, there are not 4, but 5 taste sensations that those fungiform and circumvallate papillae are able to perceive. And those are, of course, sweet, salty, sour, bitter. Those are the 4 that most people are aware of. But there is also a savory sensation, or again, because it was first discovered by a Japanese researcher, what they called umami, which is that savory sensation. And these are the 5 taste sensations that we're able to perceive. Now, if you go, and again, it's a little better now than it used to be back in olden times, but in olden times, when you would go into a kindergarten class, right, when they'd be learning about the senses, there'd be a picture of the tongue on the wall, and there'd be a big circle around this part, that this is the sweet part. That's why you lick ice cream cones, because this is where all your sweet sensations are. And salty's over here on the side, and bitters in the back. And we had these really precise taste maps, right? And we learned about these in elementary school. And it's not just elementary school where you hear about these as well, right? Again, back in ancient times, you used to be able to leave the house and go shopping. And what, excuse me. And one of the places you might want to go shopping at is Williams-Sonoma. If you were to go shopping at Williams-Sonoma, among the many fun things that you can buy there is you can pay a money to buy a pair of glasses made by a company like, for instance, Riedel. Riedel is one of these famous companies that makes specialized wine glasses. Again, a single wine glass, it can be upwards of $20 to $24 for just a single wine glass. The cheap ones are like $14 to $16 for a wine glass, but they're worth every single penny. And the reason for that is they have the perfectly shaped bulb on them so that as you swirl your wine, it adds just the right amount of air to it to maximize your enjoyment. But what really makes it an engineering marvel is that the lip of it is also set in such a way that as you pour the wine into your mouth, it presents the wine to your tongue in just the right order, in the right locations, so that it hits the right taste sensations at the right time to maximize your enjoyment of the wine. And all of that sounds incredibly amazing, and it is complete bullshit. Right? It is not, it is completely ridiculous, completely untrue. Every single one of us has a taste map. In fact, that would be a great activity for you to do while you're trapped at home. What you can do while you're trapped at home right now is you can do your own taste map. All right? The way you can do this is to have a glass of water present, have a paper towel present, and what you do, rinse your mouth, dry your tongue with paper towel, and then what you are going to do is you are going to take a little glass of lemon juice. And what you're going to do is you are going to take a Q-tip, dip it in the lemon juice, and then you're going to slowly move it back and forth over the surface of your tongue. And what you'll find is you don't taste the sour of the lemon juice over all parts. Maybe you'll sense it here, and then you'll sense it again here, and you'll sense it again here, and you'll sense it here, and you'll sense it here. Then you rinse out your mouth, and instead you get a glass of salt water. Dry off your tongue, and take a Q-tip with salt water, and you'll see salty here, and salty here, and salty here, and salty here. And then you'll do the same thing with sugar water, and you'll get that. Coffee is always good for the bitter. If you are not sensitive to it and you happen to have it in your cooking arsenal, MSG is one of the great things that triggers that umami. That's why they put it in a lot of Chinese food, is because it makes it much more savory and much more enjoyable and you want to eat it. Problem is a lot of people build up an intolerance to MSG, so if you eat too much of it, you get major headaches and other problems with it. If you're a carnivore, you can use the juices from red meat. When you cook red meat, you get those red juices that come out of it. That can provide that sensation. If you're vegan, I have no idea what produces the umami sensation that is safe that way other than MSG. So again, but you can do the other four fairly easily and safely. And what you'll find is you will indeed, mushrooms, there you go, exactly, especially cooked mushrooms. Yes, soy sauce, that's good, excellent. Soy, that can do that. Absolutely, you guys can, excellent, good examples. Especially portobello mushrooms give you much more of that umami sensation cooked. So those are really good. The juices from that are excellent. Great example, I like that. So again, I didn't have time because I was grading your test to make this as an activity. Maybe if I have to do this again online, I will make that an activity that you guys have to do at home, because that's something that can be pretty simple at home. And you have to draw your taste maps and turn them in. But do that for yourself, do that for your family, and you'll see that everybody's is different. Everybody has a taste map and everyone is different. Notice also the one thing we have not talked about is spicy. All right. What you have to remember is this taste sensation involves chemoreceptors. Right? We are, here's a fun fact. These are chemoreceptors, where basically what we are experiencing is a chemical dissolved in water. And that chemical dissolved in water binds to a receptor, turns that lock, and when it turns that lock, we get that signal of that sensation, sweet or sour or anything along those lines. What's really cool about that is obviously it's not truly dissolved in water, it's dissolved in your saliva. And the components of your saliva are actually different from the components of others. One of the studies they've done is they have taken saliva from an individual and saliva from a second individual and dissolved the same amount of a chemical inside of them, and then wash them on the surface of somebody's tongue. And what they found is people are much more sensitive to chemical perception in their own saliva than they are in somebody else's. right? So there's no benefit to swapping spit with somebody. Your own saliva, your body, your nervous system is familiar with, and so we're actually more critical, better able to distinguish chemicals that are dissolved in our own saliva than somebody else's. All right, so there's that. Again, if you may not have thought of it in these terms, but I know you're aware of it because any kind of diet, something that has been made, like Diet Coke is a great example, they have tried to find a chemical that mimics that binding of the glucose, right? If we can get that magical chemical that binds to the glucose receptor and turns that signal, you get the sensation of sweet without the calories of the sugar, right? And so that's the goal of all those diet drinks and things along those lines. But that's where spicy comes in. Spicy primarily is what component? What is the chemical that we perceive as spicy primarily? There are many, but what's the primary one?
Capsaicin.
There you go, capsaicin, absolutely. Capsaicin is the janitor's key. And what does the janitor's key open?
Everything.
Everything, exactly. So what happens is when you eat that spicy food with capsaicin, that capsaicin binds to all the receptors and turns all those keys. And so we get a massive neural signal sent to the brain. And that massive neural signal sent to the brain is perceived as pain. The other thing that capsaicin does is it simulates the temperature receptors, tricking your tongue, your mouth, your lips into believing that they're being burned, that the temperature has gone up. And of course, as we know from homeostasis, when temperature goes up, blood flows to that area to radiate that heat away. So that's where when you eat those spicy wings, your lips get all red, your face can get all red, you get all sweaty, all of that because it is tricking you into that pain sensation, that heat sensation. It tricks all those receptors and then causes that, that painful experience. And where else do we have those types of sensitive receptors? In the anus as well. So you get to enjoy those spicy wings a second time as that capsaicin leaves the body as well. All right, so there you go. Those are our taste sensations. What is next? Our tongue has muscles in it. We learned about this a little bit when we got in our nervous system. We talked about it when we were talking about our cranial nerves. Basically, our tongue, which is a big, huge muscular organ, has two main types of, let me use black for starters, two main types of muscles. The first are the intrinsic muscles. Intrinsic muscles are muscles that are contained completely within the tongue. And it has fascicles, unlike any other muscle in the body where most of the fascicles are more organized. Here, they're very disorganized. They're all over the place. And the intrinsic muscles are what allow for us to change the shape. So if you can roll your tongue or make your tongue flat or round or curl the edges or flip it over, that ability to change the shape of your tongue is the function of the intrinsic muscles. Extrinsic muscles start in the tongue, but they actually exit the tongue. And as they exit the tongue, they primarily connect to that hyoid bone. But there are other structures they can anchor to as well. And the function of our extrinsic muscles is to move the tongue in space. So your ability to raise your tongue to touch the roof of your mouth or stick your tongue out or move it left and right, that ability to project your tongue and move it through space is the function of the extrinsic muscles. So there are both of those. And of course, as you know, both of those are controlled. The extrinsic and intrinsic muscles are, of course, controlled by cranial nerve 12. And cranial nerve 12 is, of course, the hypoglossal nerve. All righty. We talked about the sulcus terminalis already, basically that dividing point between the circumvallate and the lingual tonsils. We talked a little bit about the lingual frenulum. Again, the lingual frenulum is what anchors the tongue in place, giving it a limited range of motion, but a fairly important range of motion. And again, you can touch your tongue to the roof of your mouth. You can move it around to produce speech. You can stick it out. You can do all those types of things. However, in some instances, the lingual frenulum is too short. And if it's too short, the tongue can become too restricted in its movements. This obviously can affect things like speech. But the other place where it can cause a problem, especially in infants, is if the lingual frenulum is too short, then the baby can't latch on properly, can't use its tongue to latch on and get suction to be able to express the milk from the breast. Of course, there is a common term for that, and the common term for that is literally to be tongue-tied. So the condition of having a lingual frenulum that is too short is commonly referred to as being tongue-tied. And how do they resolve that if you're tongue-tied?
Surgical.
Yeah, surgical by doing what?
By cutting it.
There you go, exactly. Basically, they just go in and they cut the lingual frenulum, right? Usually, again, that's done for that case of being tongue-tied, having that condition. There's speculation that Gene Simmons, for instance, had his cut on purpose or tore it at one point. There's speculation both ways, which is why he's able to project his tongue so much more prominently than other people are able to do. Of course, saying that someone is tongue-tied is fun, but of course there has to be an appropriate anatomical term for that, right? Something that you're almost certainly going to have to spell on an exam at some point. And so, of course, the correct anatomical term for having too short of a lingual frenulum is the condition of ankyloglossia. I know tongue-tied is much easier to say, but it will not be acceptable on the exam. We need to use appropriate anatomical terms, so know how to spell ankyloglossia, which is that condition of having too short of a lingual frenulum. Again, our tongue lined with a non-keratinized stratified squamous epithelial tissue, lined with a mucous membrane, produces mucus, but as we also talked about, it also produces that lingual lipase, which again, in an inactive form, which will become active in breaking down fats in the stomach. All right. Questions on that? All right, the next accessory organs that we need to talk about, and how are we on time? We're doing good. Okay, this will be the last thing we talk about. I am not going to have the opportunity to go too much in depth into the teeth. Make sure you look at the teeth, look at the anatomy. I think the teeth anatomy is pretty simple and straightforward, so I don't think it requires too much from me. I would much rather, and I think it's much more efficient to use our time this way on the salivary gland. So to stay on pace, because again, we lost a day. So we only got 3 days to talk about this, is going to be for you to take the onus of the teeth, because I think it's pretty simple and straightforward, although we will talk a little bit about it at the beginning of next class. But let's make sure we can appropriately get through this part, because this part is the more challenging part, I think. And that is our next accessory structure, and those are the salivary glands. Notice we have 3 pairs of salivary glands. So technically you have 6 salivary glands, 3 on each side of your oral cavity. And you have 3 different types. Here we see them in this great picture from your textbook, where we're able to see the 3 glands. Here we have the largest, which as you can see here is the parotid gland out here. Notice it is the most superficial. It is on top of the masseter. There's our masseter. There's our buccinator muscles that we can see there. Basically, it sits right underneath the zygomatic arch in front of the ear. The second is the sublingual gland. It is just deep to the tongue, underneath the surface of the tongue. And deep to that, and a little bit more posterior, located down here deep in the mandible, is, as the name would indicate, this submandibular gland. Not only are these glands different in their location, notice again, these are all exocrine glands. So that means they have ducts, and their ducts express into different locations as well. Notice our parotid gland, because it's so superficial, its duct actually goes over the masseter, passes through the buccinator and then releases its secretion into the vestibule, just where the second molar is located. So here are our 3 molars. At the second molar and the superior vestibule is where our parotid duct releases its secretion. Notice our sublingual has not one but many ducts that all line up along the lateral aspect of the tongue. So if you look at the underside of the tongue, you can see them on the lateral aspect. And let's cheat and go back. Notice if we go back to this picture here, those are the ducts for our sublingual. But notice right at the edge where the frenulum is, there are 2 paired ducts as well. And those 2 paired ducts are not actually from the sublingual. That is the singular duct of the submandibular. So again, these larger ducts here are the submandibular ducts. These here are the sublingual. And then while we don't see it, right up here in the vestibule of the second molar in the superior vestibule, next to the second molar, is where the duct of the parotid would be located. Different locations, well, different glands, different locations, different ducts, and where they express is different. And the other way they're different is the substance that they produce. Collectively, obviously, they all produce saliva. And let's cheat and I'll put this on here. Collectively, they produce saliva. But really, saliva is primarily made of 2 components. The first of those is serous fluid, right? This serous fluid is the part that contains the water, making it watery. This is the one that contains the enzymes and the antibodies and all of that stuff, etc., that is going to be in there. And again, its job is to help to moisten and provide protection that is in here as well. It's a watery component. However, any of you that are older siblings, if you're an older sibling worth your salt, at some point or another, you have kneeled on the chest of a younger sibling and dribbled the tiniest amount of saliva out of your mouth, dribbled it down in a long stream so that it almost touched their nose, and just before it touched their nose, you sucked it back up. And the reason you were able to do that is because saliva is more than just serous fluid. It also has a mucus. Oops. And of course, mucus, we know, is formed by a protein. Anyone remember the protein that makes mucus?
Mucin.
Mucin, there you go, excellent. So we have some mucus producing, or really mucin producing cells. And then of course, once that mucin is hydrated by the serous fluid, it becomes mucus. So when we look at these 3 glands, not only are they different locations, obviously have different names, they have different duct structures, but they're also going to produce a different composition of the saliva as well. And we can know that by looking at them, and we can also tell that by looking at them histologically. All right, and let's see that in just a minute. So let me go ahead, I need to clear that. Let's look at our 3 glands. Starting first with the parotid. The parotid, as I mentioned, is the largest and most superficial. So again, it's just inferior to the zygomatic arch. It primarily produces a serous secretion. Like I said, that serous secretion is going to have water in it. It is going to have the antibodies. It is going to have the enzymes. And if we remember anything about our structural classifications of our glands, and for that matter, functional, well, you need to talk functional as well. Actually, let me do this way. Hold on, let me delete that. For any gland, we need to know ever, but especially here, there are 4 things we need to know. Obviously, we need to know its name, right? We need to know the substance it produces. And we also need to know the functional and the structural classifications. All right. So this is something we need to know for all of our glands. Here in this case, we are dealing with the parotid. And obviously it is going to produce saliva. We'll talk about the composition of it in just a second. The good news is for every single salivary gland, the functional classification is the same. All of them, all of our salivary glands, oops, if I spell all correctly, all of our salivary glands use the merocrine mode of secretion. Anybody remind me again what that means? What was the merocrine mode of secretion? Guess who gets to all go back and look back at their structural and functional classifications of their glands. Merocrine basically means they use exocytosis. They produce this secretion, bundle it into a vesicle. That vesicle comes to the plasma membrane and is released via exocytosis. The cell is unchanged by that process. It's not damaged or killed, so it is something that can be done continuously. All right? Now, when it comes to structural classifications, as I'm sure you remember, and it doesn't sound like any of you do, so you get to go back and look at structural classifications too. There are two main types of secretory structures. In those two main types of secretory structures, some of them are ball shape, and those ball-shaped secretory structures are called acini or alveoli. Both are acceptable. The other are long, thin shaped secretory structures called tubular secretory structures. We have two different types of secretory structures, and conveniently enough, in saliva, saliva produces two main types of substances, serous fluid and mucin. As it turns out, in our salivary glands, in all of our salivary glands, it is the acini that produce the serous fluid, whereas it is the tubules that produce mucin. So when we talk about our glands, and in this case, we're talking about our parotid gland primarily produces serous fluid. So based on that, what would the structure of its secretory structures be? Shape, let me say that again. Shape. Yeah. Secretory structures is, oops, did I spell that right? Is going to be acini, right? Because it is producing serous fluid, small, tight, ball-shaped structures. And being as big of a gland as this, is it going to have just one ball-shaped secretory structure?
No.
Or just one duct that all of these ball-shaped structures are going to feed into? No, of course not. It's going to have two or more ducts. It is going to have two or more secretory structures. So when we talk about its structural classification, for our parotid gland, it is going to be a compound. Oops. The acinar, or compound acinar, is its structural classification. So when we talk about our parotid gland, it is going to produce saliva, and now we know a watery saliva. Its functional classification is merocrine, and the good thing is that all of them are merocrine. So if I show you any salivary gland, and I ask you to identify its functional classification, boom, that's easy. And in fact, any of the exocrine, let me phrase that, all of the exocrine glands of our digestive system all use the merocrine mode of secretion. So pretty much any exocrine gland that I ask you the functional classification, you know the answer. It's always going to be merocrine. And here, its structural classification is a compound acinar. All right? We can actually see this if we look at it histologically. Let us, crap, am I going to lose this? Let me do this. Can you guys see that? Oh, here, let's do it this way. How do I want to do this? All right, I'll do it at the end. That'll work. I'll just do it at the end. All right. We will look at these things histologically at the end to make sure. So it's going to be the structural shape is acini. And again, these are going to be small, dark, ball-shaped secretory structures. Because I don't want to lose, I want to keep this. All right. So here's our parotid gland. We've identified what it produces, and as we talked about, its duct releases its contents into the superior vestibule next to the second molar. And again, notice it says vestibule, but make sure you say superior vestibule because we have two vestibules. All right, questions on that? All right, let's do the same thing with the sublingual. For the sublingual, and we're going to have to cheat, I have to put a line through this so that I can type instead, and we'll do it in blue, sublingual. Oops. Our sublingual gland. Sublingual gland produces, instead of a watery, it produces, hold on, delete that, erase that. Oh no, not that one. I really miss my whiteboard. Our sublingual gland produces primarily mucin or mucus, right? Is what it primarily produces. Based on that and what we know, since it's producing mucin, its shape of its secretory structures are going to be tubules. Now, if we remember back to the skin, remember when we learned about the sudoriferous glands? Sudoriferous glands were simple coiled tubular glands. And when we looked at that under the microscope, did we see that one continuous like string of tubules through the whole thing when we looked at it under the microscope? No, remember, because you have a three-dimensional structure that you're slicing through. So if you have a ball of yarn and you cut that ball of yarn in half and look at the cut edge, you don't see one continuous piece of string. You see a whole bunch of little cut pieces of string at different angles, in different directions, at different shapes. And that's what happens when you look at this. Even though they're long tubes, when you cut them, you see all these irregular shaped pieces when we look at these mucus tubules. All right? And so when we look at these mucus tubules, it is going to be structural classification then is going to be a compound tubular. So we're going to have these compound tubular glands. Notice there's one other way you can kind of tell what these glands are doing, and that is to look at their ducts. Notice the parotid gland is way back here next to your ear, and it has to release its secretion way up here in your mouth next to your second molar. A long, single, narrow duct. If this parotid gland was producing a thick, viscous mucus, would it be easy for that mucus to travel that long distance to get into the oral cavity? No.
No.
But that watery serous fluid has no problem flowing through that. Notice our sublingual gland, which primarily just produces mucus, has short ducts and has many short ducts. so that any of that mucus doesn't have to travel very far to get into the oral cavity. So it has many ducts, all very short, because it's producing a much more thick, a much more viscous secretion. And that much thicker, much more viscous secretion, that mucus is going to have to travel a shorter distance. So again, we did all of this, but it is on the floor of the mouth under the mucous membrane, produces mucus secretion, has that buffer, provides that lubrication to it, right? Greases the wheels so that it can allow swallowing to occur. And then we have those sublingual ducts, multiple sublingual ducts that come up lateral to the frenulum. And again, I don't want to erase what we have on that, but here we see that picture. So again, up here, next to the second molar is where our parotid gland, and then these to the side is where the sublingual comes out. Our third is our submandibular. So let's change colors and fill out our chart one more time. So get rid of that, get rid of that, get rid of that. Oh no, that works perfect, excellent. So we'll use green this time. So now we are talking about the submandibular gland. Notice the saliva it produces isn't all mucus, isn't all serous fluid. It produces both mucin and serous fluid. It's still going to do it using the Mericrid mode of secretion. But when it comes to our structural classification, it is going to be compound because it's going to have multiple secretory structures and multiple ducts. And again, if this is all confusing to you, go back to the tissues section. I think that was chapter 4 when we talked about epithelial tissues. In epithelial tissues is where they talk about exocrine glands. Make sure these terms, structural classification, functional classification, merocrine, holocrine, all of these things mean something to you, because it's not just the salivary gland. Every gland we talk about, you're going to need to understand these exocrine components. This one produces both mucin and serous fluid. That means it has to have both tubules and acini, and that we see in its name. Structurally, it is a compound tubuloacinar gland because it has both tubes and acini, because it produces both serous fluid and mucin. And it releases its secretion, as we talked about, right next to the lingual frenulum. And I think I have that pretty picture here again. Perfect. This location right here. You may not have ever thought of these salivary glands in this way before, but maybe you or someone you know has the ability to be able to, with the right kind of contractions and movement of their jaw, be able to, oops, wrong button, express saliva out of these submandibular glands. What do we commonly refer to that as?
Gleeking.
Yeah, there you go, gleeking. I don't know, there may be a technical term for it, but I don't know what it is. But yeah, that ability, that gleeking, that ability to express it, that's basically what you're doing. You're contracting the muscles in your neck and in your jaw, moving your jaw movement in a way to basically squeeze that submandibular gland and release its contents into, or express it out of the oral cavity in that fashion. So using your tongue and whatever else it is, I'm not able to do it. So however you do it, that's what that is. And so again, that is that. All right. Now notice we have talked about all of this. And now that I've got it on the board, I want to switch now to, let's start. Where do I want to start? Where do I want to go? I want to go here. Here's some nice histology, but let's again talk about how I got there. From there, I went to my study area, right? From my study area in your modified mastering A&P, and again, I know the way you get there is slightly different from me, but here, under lab tools, we have this practice anatomy lab. And when you go to the practice anatomy lab and pop it up, one of the things that they have here is histology. Notice now we are looking at histology of a salivary gland. Let's, hold on. There we go. I think I want to go back a picture. There we go. Notice here, when this gland, and this happens to be, right, spoiler, the parotid gland. Notice we see many ducts on this. Well, that's a blood vessel. Many ducts on this, and a ton of secretory structures. And as we could do the plus and minus here, but we'll just do that simply. Notice when we look here, we clearly see a duct, and we see lots of these dark ball-shaped secretory structures. And they're as far as the eye can see. These are all serous fluid producing acini. So this is that acini, and it produces that serous fluid. Here we see it at a high magnification where we can see the acini very clearly. But notice we come back here, right? We see plenty of ducts. We see lots of secretory structures, but they're all fairly uniform in shape and size because they're ball shaped. And pretty much however you cut a ball, the ball pretty much looks the same. Look how different this looks from this. I think if I go one more. There we go, no. Yeah. Notice here, the secretory structure, we still have ducts, big ducts that we can see. But notice now the secretory structures are much larger, much more open. Notice as we go through the pictures, here we see one in a big, long cross section. Here we see, a longitudinal section. Here's more of an oblique section here. And they're big and open. Remember our goblet cells that we talked about when we were looking at our mucous membranes have a lot of mucin in them, and they tend to be more clear. So I have these clear, pillowy, more irregular shape secretory structures. Here's a great view of them. Again, plenty of them are in cross section, but we also see some longitudinal sections through these things as well. So this is an example of our sublingual gland, all mucous tubules. The parotid gland was all serous acini, and we can clearly distinguish that histologically. But remember, our submandibular is a mix of both. Notice here, we're going to see three things. We're going to still see our ducts, because these are exocrine glands. They all have to have ducts. Notice over here, we see our dark ball-shaped serous acini. So we see some of those dark ball-shaped serous acini. But notice we also see some of those light-colored mucus. That's not a mucus acini. It's a mucus tubule. I got that wrong. These are mucus tubules. And especially if we go back to the next view. Right? So we can see ducts. We can see our serous acini, and we can see those clear mucus tubules in these things as well. So we get a mix. Notice it's not as uniform looking, and again, they've got them labeled wrong. They're not mucus acini, they're mucus tubules. But I didn't realize that when I looked at this the first time. But we can clearly see the difference in them. Notice here, you can clearly see that this is a big tube-shaped structure. Yes, that is a cross-section, but that's just a cross-section. That's not an acini. These are just tubules at irregular shapes. So we can see ducts, serous acini, and mucus tubules. And this is a compound tubulo-cinar gland. All right, so notice the uniformity of the first two compared to the lack of uniformity in this. This is a mix of both acini and mucus tubules. Here is all mucus tubules. Here is all, and here in our parotid is all serous producing alveoli. So histologically is how you're going to be able to tell these three apart from each other. You should be able to identify all of them histologically and distinguish them from each other. And again, we got to this in the practice anatomy lab, right? Another good place, remember to go to, that visual anatomy lab. When you open this visual anatomy lab, They've got a digestive system. And when you look at the digestive system, they have some histology. They have some digestive models and charts that are really, really nice, including, all right, again, this is exactly the picture that we had on the last exam, showing the fosses, the space in between, showing the two arches, showing that tonsil, right, frenulum, the gingiva. So we see a lot of these structures. They have a lot of these great pictures in here. And here's that other picture. Notice here are the two arches. Here is, again, that arch coming from the roof of the mouth, palatoglossal arch. And this one coming from the roof of the mouth towards the esophagus is the towards the throat, palatopharyngeal arch. And there's our friend, the trachea. Notice there's our epiglottis. Not our trachea, our friend, the larynx. There's our epiglottis, aryepiglottic fold, cuneiform cartilage, corniculate cartilage, so we can see all of that. And then way down there is our glottis. So there's some great things. And then obviously this back here is our oropharynx, which leads down to the rest of the laryngopharynx, oropharynx, laryngopharynx, and then down into the esophagus. Here, we also have the circumvallate papillae. So again, notice there's a lot of really, really good things on this. So take advantage of these resources. And then, like I said, it's got a lot of things. It doesn't have everything, but it has a lot of really good things, and we will do even more. All right. Back here to finish this off. So these 6, really, 3 pairs of glands work together. Oops, I lost that. There we go. Work together to produce on average. And again, one of the things that I'm going to say again and again and again during this section is these numbers are averages. They change dramatically based on 3 factors: your frequency of eating, the volume of food you eat, and the composition of the food you eat. All of those 3 factors affect how much of each of these secretions you produce. But on average, we produce about 1 liter to 1.5 liters of saliva each day. Again, the majority of it is actually produced by that mixed gland, the submandibular gland. Saliva is mostly water, but like I said, it has electrolytes, immunoglobulin A's, lysozymes, defenses, enzymes, mucins, buffers, all sorts of other things to facilitate in the digestive process, right? Maintain the moisture, maintain the integrity of our oral cavity, protect us from pathogens from the outside world, and mix with that food, produce that bolus, that first iteration, that first change that our food undergoes. These salivary glands are part of our rest and digest process, so they are primarily controlled by our parasympathetic nervous system. So parasympathetic stimulates, all one word, parasympathetic. And if you remember correctly, it was cranial nerves 7 and 9 that are involved in innervating them. So our facial and our glossopharyngeal are the ones that are going to innervate these 3 glands. From the sympathetic, because again, when you get really nervous and you have to give a speech in front of people, your mouth dries up, and that sympathetic pathway, remember, it is going to be a sympathetic nerve. And that sympathetic nerve comes off of primarily let's just say it this way, cervical chain ganglia. So those are the 2 nerve pathways that get us to our salivary gland to either increase or decrease. And again, that's the key. Parasympathetic is going to increase saliva production. Sympathetic is going to decrease saliva production. Excellent. All right. So like I said, I'm going to skip the tooth part because I want you to work on that on its own. It's pretty simple and straightforward. And we've gone about as far as I wanted to go for today. So this is a good stopping point. We'll go ahead and stop here, and we'll pick up briefly, quickly talking about the teeth in the next one, but then diving straight into the new material because we got a ton of stuff to cover. All right. Questions on any of that? All right, that is a good start. Now, again, one of the things that I want to remind you is that we are starting this class an hour later than we normally would. So I know our class normally runs till 11:35, which we're past now, but remember, we're also starting an hour later. Wednesday, my goal is to cover a lot of material because I want to cover as much as I can before the exams weekend. When you have that exam, I don't want to cover it as much 2 days before the exam. So I am going to likely go beyond where we are here, get closer to our normal 4-hour lecture to make sure that I can cover as much as I want to. So expect to go late, to go long. And again, it's not technically long because we're starting an hour later, but I will be talking for more time in the next class, so be prepared. All right, questions for any of that? All right, hopefully you did your pre-lab and turned it in. If not, please make sure you get it done and turn it in. Partial credit's better than no credit, but make sure you got that. You should have gotten that notification. You should have gotten that done and turned in. All right, it was due at the beginning of class. All right, guys, thank you very much. I hope you had a good weekend. Like I said, continue to be safe, study hard, and I will see you on Wednesday. Wednesday, I will have the grades out, and we can talk about grades. We can talk about reviewing your exams and all the fun things that'll go along with that. All righty. Take care. And any questions before we call it a day? All righty. I'm going to go ahead and stop the recording. You have a good day today.